Well, good afternoon. We hope you are back with us. We hope you've enjoyed your lunch. We've had a great lunch and some time of fellowship here, again, at the 2024 SPF Annual Conference here in Tampa, Florida. Right now, folks are enjoying some time together, getting to meet new friends, visit with some old ones, and I've got a new friend that I've just met here today for the first time. I want to introduce you to Johannes Sauer. He is with Cadence Shoe Company. And Johannes, just from looking at these shoes as you showed them to me earlier, those shoes are pretty amazing, and I look forward to trying a pair. So let me just ask you to share with our visitors at home, share with them about these shoes, because all of us, you know, there are probably 125 of us here who've got one of these conditions. And probably all of us walk a little differently. You know, how we get impacted and affected by the muscular and the neurological issues is different in everybody. But for me, I can tell you the way I walk, I'm a scooter.
Yep.
And what you're going to tell us about these shoes would help a guy who's a scooter. So share with us about yourself and about this company and about these shoes.
Will do. Well, first of all, Greg, thanks for having me down here at the conference. It's an honor to be here, and it's so fun to see everyone and have conversations just like this one here. My name's Johannes. I'm the CEO of Cadence. We're a brand new startup based in Santa Barbara, California. And we've made the world's first truly adaptive shoe. Shoes made for people that are experiencing walking difficulties. So think anyone that shuffles, hikes their hip up, throws their leg out to the side, really anything when you worry about tripping and falling, the shoe could be really good for you. And the way it works, it has two distinct areas, but before I get into that, I'll tell you a little story on how it started, because I think it's fascinating. My co-founder, Dr. Tyler Sasko, started this as his PhD project at MIT, and it was a big robot that was made for people post-stroke to rehab and to learn how to walk again. And it was two treadmills that would basically mimic removing the floor as you were walking. So what people found was it was a lot easier to walk, they would normalize their gait, they would pick up some speed. And then one of the study participants was like, man, I wish this wasn't $1 million and I could take it out of the lab. And, you know, fast forward 10 years later, here we are with some really nice looking shoes that, you know, help people to walk better.
That's a fascinating story. It's good to know how things can happen that next year or 3 years from now have us working in a very different direction that makes a lot of difference for a lot of people, and I think these shoes would. Again, we all walk differently. I'll tell this quick story. In our annual conference, I believe it was 2019 in San Antonio, a former board member, and she may be listening today, a young lady named Jean Chambers from the Pacific Northwest, shared with me as we sat there visiting, we were talking about walking problems. She had on a pair of shoes that she had resurfaced on the bottom all the way up to the toe with a slicker surface. And she said, here's how you do this, go to your shoe person. So we found our shoe person at home. I bought 3 pairs of shoes specifically for that, took it to him, and now those are the shoes I wear every Sunday when I go to church. Why? Because they help me get from point A to point B.
There you have it.
Now share with us, you mentioned that the shoes might be varied for different walking issues. Share that with us.
Yeah, so basically what the cobbler did for you is we've built this into the shoe. So for the viewers at home, you can probably see there's two on the outside of the shoe sole, on the top part, there's 2 distinct areas. Those are nylon pucks. And nylon has low friction properties, meaning it slides. And it's raised off the bottom of the shoe. And in the middle part of the shoe, there's rubber, which is sticky. So what the shoe basically allows you to do, think a door gap, low pile carpet, anything that would typically worry you for tripping and falling, you now slide over it. But you don't want to be always sliding, right? Because that would make you feel unsafe, unstable. And so as soon as you put weight in the front of the shoe, the middle part, the rubber part, comes down and you can stop and toe off. So in a nutshell, the shoe slides when you want it, but it's sticky when you need it.
And for some of us, we have a tendency to drag a toe when walking. Yep. Are there any options now, or are you guys planning anything in the future that might solve that on the end of the shoe?
Yeah, it's a fantastic question. So you can also see it's shaped like a rocker. So already you should have an easier time walking, but we are in the process of adding, you can basically buy a little part that will go on the tip of the shoe, and that will then have additional nylon for you where you don't have to worry about clipping your toe anymore.
Can you share with us two things, what the price of the shoe
Yes.
is, and if someone was interested, how would they look into getting a pair of your shoes?
Yeah, great question. So it's $198, and if you think about it, a pair of Nikes costs $150, right? But Nikes don't have any adaptive features unlike our shoes. And you can just go to Cadence.com, that's spelled CANDESE.com, and get your pair there. The great thing about that is you can try the shoes in the comfort of your home, right? And you have a 30-day risk-free trial with that. So if the shoes don't work out for you, and listen, while I hope it works out for a lot of people, I realize it won't work for everyone. It's just the nature of who we work with and cater to and serve as customers. It's not going to work for everyone, we realize that, but we want you to try it. And so there's a risk-free trial with that, so go to our website and order it from there.
Well, when you talk about the price, I know $198 may sound like a lot, but if you've got a grandson or grandsons like I have who are athletic, and when they call Granddaddy or Gigi, can you get me this pair of shoes? And $198 sometimes would be a good deal on the shoes they want. So those shoes can be very expensive, and the idea that a shoe like this might make my getting from point A to point B easier, and more importantly, safer.
Yeah.
You know, because I decided a number of years ago, actually Norma twisted my arm. I didn't want to go to a walker or to a cane. Leave me alone, I can walk. And what she convinced me is, Greg, if you trip and fall one time and you break an arm or a leg or a hip, you may not recover like you need to recover. So everything we can do to be safe and careful in our mobility is extremely important. And I'm excited about these shoes. I can't wait to learn more about them for me. Johannes, anything else about these shoes or your company that I haven't asked you that you'd like to share?
Well, a couple of things. So I think what we haven't talked about is it's easy to get on, and I'm holding the microphones, it's hard to demo, but so it has a Velcro. While it looks like a lace sneaker, these are just for design, so it's really easy to get on and off with the Velcro. Beyond that, what our customers tell us, and of course taste is something that's, you know, everybody has got a different opinion of what looks good, but I think you can't really tell this is a sneaker that's designed to help with walking. We made sure it's a product that looks good, and so, you know, we'll keep coming out with different colors, styles, and all of that, but rest assured that any product that we make is gonna look good and make you stand out less, and you know, that's what we care about. We really want to make sure that you have a product that you are proud to wear.
And if I want to try those, I just want to emphasize again, I can order a pair, and I've got a 30-day time frame to determine if they work well enough for me to keep.
That's
and
right.
If not, I can send those back to you and get all my money back.
100%.
That you can't beat that deal.
No, it's as it should be, right? We want to make sure that the money that you spend on something works for you, and you know that everybody's happy with the product.
Well, Johannes, we want to thank you again so much for being here, for your company being one of our sponsors and present in our hallway and visiting with people. And it's been curious to watch as you do that. I haven't looked down the hallway many times that someone hasn't been there at the table with you. So a lot of folks are learning about these shoes, and that's good for them and good for you as well. And we appreciate your involvement, your support here with our conference. And we will continue to make people aware of this opportunity best we can. And you can rest assured when I get home, I'm going to be checking them out.
Awesome. Thank you so much for having me. It's an honor to be here. What a great conference. I'm so impressed. I'm seeing a lot of these days, and you guys have put together a fantastic conference, and all the people here are so lovely. So thanks for having us.
Thank you for being here with us, and we'll let you get back to your booth and maybe sell a pair of shoes or two.
We'll do. Awesome.
Right.
Thank you so much.
Well, guys, again, there's a lot going on with us. We appreciate Johanna sitting in. If you could be here, we've got probably more vendors visiting with us in the hallway. They're sponsors. They've helped support financially the conference. They've got a number of products that we can look at that might make our lives a little easier, a little more mobile. So look at our program and look at those possibilities. And if you have a walk like I have, at home I'm still up on a cane all the time. I only get in a chair when I go out in public, and that's for safety purposes. But there are places I go that a shoe like that, if it could help me scoot along much more safely, would be very important for my health and my mobility. So you guys might take a chance to look at that and see if a pair of those shoes might help you. I'm trying to bring in here now, my next guest is going to be Kim Sanchez with Social Media Podcasts. One of the things our marketing committee's been working on these past few years is how to get the word out much better about the foundation, about the fundraising that's necessary for us to fund research, about social issues that our population needs to know about. A lot of what we do here at this meeting is get to know one another, share life stories, share experiences so we can make life better at home. We also try to do that all year long through things like SPF talks, through the email blasts that we do, through the synapse letters that we send you. But part of that marketing involves more of a social media context than ever before. And Kim is going to come talk to us about how she does that and how she can assist the foundation in doing that much better. She's on a little scooter as well, so give us just a moment to help get her in here, and we're going to introduce you to her. Come right on around here, dear.
Would you prefer to stay in the studio?
I'll give the chair. Yep.
Give Kim just a moment to transfer. We're all a little bit slow, but we'll make it. Come on, Kim.
Got it. Just make sure you don't tip. Okay. There we go.
As I watch you transfer, that's about how I transfer, so we might make a good dance team. Who knows?
Oh yeah, for sure.
And while you've been sitting there for a moment coming this way, I've been telling everyone about you.
Awesome.
And about the podcasting and the social media kinds of things that you do.
Yep.
And how you've agreed to help SPF and our membership learn how to do that better.
Yep.
In fact, if you were here with us, Kim's going to have a spot outside where any of our folks will stop by and visit with her. She may do a podcast right there, or at least show them how to do that.
I've done a few interviews.
So tell us
already.
about it all, Kim.
Just in general, I can tell you guys I have PLS. I was diagnosed about 2 years ago. I started off doing an Instagram where I wrote kind of things about my journey, just almost like a journaling, right? It was like very therapeutic to just share my experience. And it was actually my brother who said, you should think about doing TikTok or Instagram. And I thought, I'm not that kind of person, right? I'm not out there sharing my story kind of a person. But I did start doing videos, which I think, today kind of catches on more. People don't like to read as much as they like to listen to little sound bites. So about a year ago, I started doing a TikTok and Instagram where I shared my story. And it kind of started off around ALS Awareness Month, because PLS and ALS are similar, right, on the spectrum. And I started sharing my story during ALS awareness and just getting really vulnerable about the experience and the process of getting a diagnosis. Because I know for me, I felt very alone, right? It's a rare disease and it's hard to find people like you out there. And I remember I would Google all kinds of different things. After I got my PLS diagnosis, I would Google it, and nothing comes up. Or what you do see is kind of really dark stories, and it just made me sad. So I thought, well, let me share my story and my experience, the good, the bad, right? The days when I'm feeling good, the days when I'm having trouble. And I feel like it really resonated with people to kind of find someone like them who's sharing both sides of it, right? The good and the bad.
So you started doing that. How often are you doing something daily? And if so, if people wanted to follow you, tell them how to do that on social media.
Sure. Yeah. So I have Instagram and TikTok. It's My Spastic Life is the handle. It depends. Sometimes I do it. I used to do daily. I do Motivation Mondays now, where I kind of think about something that's motivational, how to keep a positive attitude. Sometimes I share just kind of like funny stuff, and sometimes I share, stories of experience, right? Like if I go to clinic, or I go to physical therapy, or if I have a new mobility aid, or I tried the Cadence shoes that you just talked about, right? So I share kind of product reviews, things like that. So, but at least twice, 2-3 times a week, I'll share on there.
Well, you want to tune in, I can tell you, because Norm and I, as we met Kim, and we began following what you do, and it made it so much important for us to move this process along with the foundation.
Yeah.
We appreciate your help in doing that. And although it might be difficult to get used to doing it, comfortable doing it, once you do it a few times, it gets much easier, doesn't it?
It does, and I always think about, so part of, is just really documenting the story. And I know someday, my kids can look back on it and see it too. That's another thing that's really important to me. But yeah, I mean, you're kind of just talking to yourself, right? It's like talking into the mirror and just, telling your story. And there are times when, and the worst thing is, you save it in drafts and you never post it, or, maybe you redo it. But yeah, the more you do it, the easier it gets. And the more you can kind of figure out the really, what's the point of your story. And usually the night before I do it, I kind of go to bed thinking about, what do I want to talk about tomorrow? Like, what's been on my mind? And, I think that helps too for me to process my thoughts, right? Like, what happened today? Like, thinking about this conference, right? I'm sure I'm going to have lots to say about it, but think about it the night before and then just kind of, talk about it the next day. But it does get easier. And I find I get at least, I would say weekly, between Instagram and TikTok, I get 2 to 4 people reaching out to me to say, you've helped me to not feel alone. And I think especially folks who are, maybe on the younger side, right, and not knowing where to go, who to talk to, I feel different. It helps them too to kind of see it's more relatable, right? And so they reach out and say, you've given me tips or tricks of what to do, what's my next step in diagnosis, how to find groups like on Facebook is probably the biggest one, is, support groups on Facebook, things like that, so that you can find other people like you.
Well, and that's the central purpose for doing it, as you said, so that others can share the experience, so others can share lives, so we can make each other's day better. There are going to be tough days for all of us, but there are going to be days that life is good, and we need to share both of those so that we have a camaraderie in working through this process. We appreciate your willingness to help us with that, and we realize that, as you said a moment ago, some of our younger folks may be more willing, more interested in doing this, but we want to encourage all of our membership, no matter your age.
Yeah.
Any of you who might be thinking about doing this, we want to help you get comfortable with it.
Right.
And as she helps us learn how to do it, she and Norma will work together, and we'll try to learn how to circulate those things, get you guys involved in the network, and create a discussion constantly among our membership and the people who suffer with PLS and HSP, so that we can make one another's lives better and hopefully make this journey toward a treatment or a cure an easier journey.
Absolutely. And I think, I've had my daughter on my podcast talking about what it's like to be a child with a parent who has a disability. I think, caregivers, I think it's we all have that unique experience. Also, just kind of in general, people don't know what to do, right, when they see us out in public. And should I help? Should I not help? It really helps with that piece of it.
All right, thank you so much again, Kim. And you guys, she told you where to pick her up on TikTok and on Instagram, so you might do that the next day or two. And she may have some of our folks on there talking to you. So, Kim, thank you so much.
Thank you.
We're going to head toward our afternoon program with some speakers talking about how we enlarge this foundation's work. So, guys, thanks for joining us, and we will see you sometime later this afternoon.
Hey everybody, we're getting close to showtime. Okay, so we got a few minutes, so we want to make sure everybody gets tucked in, get ready for our afternoon set of presentations, okay? You got a couple of minutes to get in place. We're clearing everybody out of the lobby. Come on in, get settled in, do that stretch again, or whatever that stretch was. Get yourself ready because in a couple of minutes we're gonna start the afternoon presentation. So here we go, just hang on. Yes, yeah, please. This is not the blame for it. Oh my God. I want to take a look and we'll see what's keep an eye on. All right. All right, I'm going to do an intro.
Perfect.
First for a couple of seconds,
Great.
and I'll bring
I'll
you
get
in.
out of your way.
You can just chill with me. And I lost my camera, guys. Hey guys, we're glad that you're back in the room. I'm going to do a couple of quick announcements before we get going. This is real easy stuff. First of all, did everybody enjoy lunch? Okay, that's fantastic. Because now we're going to talk about dinner. You show you where my thoughts are at. Hey, we got written in the program we were going to do this really great thing out in the courtyard and do this kind of thing. Guess what? We ain't doing that. That's been changed up. Sorry, when we went to print with this, back in January, that was sort of the model. So here's what we're going to do tonight. When it is dinner time, after we get you through all your breakouts and everything else, we're going to have a little cash bar action out there in the lobby. Okay, going to do a cash bar at 6 o'clock. And then at 6:30, we're going to come back here and have dinner back in here. We're finding out, as much as we love you guys, and we do, we love you a lot, logistically, lifting and shifting and moving, it's getting really complicated. So we're going to keep you close. We're going to keep you inside because it's hot and it always rains in the evening here, especially where I'm at. I know that is truth. There won't rain anywhere else except where I'm at. So we want to keep you safe, comfortable, and dry, and then we'll get you dinner in here tonight, okay? Sorry about the confusion. When we print this stuff off, things change and the dynamic changes. So that's the latest update. But first of all, congratulations on being able to read the program. We're really happy that you guys are doing that. That means you're getting a lot of information because you're reading and you're keeping up with everything. Okay, well, it's that time. It's 1:30. We are ready to go with our afternoon presentation, and we're going to kick this off. This is going to be kind of interesting. We're going to have a live person here, and we've got somebody going to be zooming in too. Welcome to the world of technology. So we're going to have a really great time with this. Coming up right now is Thomas Hart from CPATH. Thomas.
Hi, everyone. Give me a second to adjust. Here we go. There. Hello, everybody. Thank you so much for being here and inviting me to speak. I actually am going to tag out a little bit, which is why we have Zoom running. So my boss, Dr. Colin Havinge, will be joining me. We are part of Critical Path Institute. I'll spend a little time talking about what that is and why we want to be a part of this discussion. And then I'll hand it over to Colin, and he can take it from there when we get into the deep science. I'm just a lawyer by training, so I've had to learn a lot about some of the drug development pipeline jargon. We'll do our best to explain all of that jargon. Feel free to find me later and ask questions. I built a flashcard deck, so we can work on it together. So a little bit about Critical Path. It was created 15 years ago by the FDA. It's a nonprofit organization based at the University of Arizona in Tucson, but we work nationally and globally. I actually live here in Florida, so welcome to my state. And sorry about the rain. So first, we're going to cover a little bit about CPATH. Within CPATH, there are different consortia, divisions, and programs. The most recent one is called the Critical Path in Rare Neurodegenerative Diseases, or CPR&D. See what I mean about the alphabet soup? Within that, we work with all of these different rare neurodegenerative diseases. So I think the Act for ALS created CPR&D, so we've started working with ALS and other related diseases. I talked with Jackie and some of your other board members about 1.5 years ago. We began our engagement, and I'm so excited to see the way SPF has grown, the work that's being done, the Centers of Excellence program, and all of you, honestly. I'm so excited to be here. After we talk about that, we'll discuss HSP, PLS, sort of that pre-consortium model that we work with, and why it might be relevant to the foundation in your work. And then we'll have some time for Q&A. Now, hopefully, Colin will be able to hear your questions. This is going to be a little bit of a trial for us. And then he'll be able to answer. So first, our mission, CPATH leads collaborations that accelerate drug development, advancing better treatments for people worldwide. That's really the focus, right? That accelerate part of it, which is a little bit different from other organizations. Wow. We go right into the dense slides, I apologize. So the way that we build these public-private partnership models is first and foremost through engagement. We're really there to be a support. We're not in charge of anything. We just lead the convening and conversation space. So that includes building consensus actively, sharing risk and cost, identifying and accelerating the best science, and working across the scientific community with the broadest experience. That way you get the input, you get the different angles, and you learn what's working in other areas that are related. It also creates this iterative model. So CPATH has close working relationships with all the regulatory entities around the world. We've called out EMA, FDA, and PMDA. EMA is Europe, FDA is the United States, PMDA is Asia. I think it's the Pacific market. I think that's what PM is. We also work closely with Health Canada and some other organizations. And that allows us to, in that pre-competitive space, identify promising practices and potentiality, move it through the drug development pipeline quickly, developing tools to speed that process, bring it in front of the regulatory bodies that need to approve that treatment, intervention, and get it to market more quickly. Maybe. There we go. Did I go? Okay, there, sorry. So lastly, these are some examples of the work we've done. I feel like it's important to know that this is a proven model. So some of the other areas that CPATH has worked in includes Alzheimer's, tuberculosis, type 1 diabetes, IBS, Friedreich's ataxia, Duchenne muscular dystrophy. I don't know if you saw the news, but just today, FDA expanded some approved access or expansion of drugs. Kidney transplants, Parkinson's, Huntington's disease. You can see the CPATH solution, and then next to it is the clinical impact. And this is where I'm going to hand it over to Colin because we've now touched on too many science words for this lawyer. So I will step back and hand it over. Colin, let me know if you can hear me. Maybe. Oh, Colin, you're still on mute. Sorry, everybody, we're live. Troubleshooting. There we go.
Hello?
There we go.
I think we're having some AV issues. Can everybody hear me?
We can hear you.
Okay. I can't see the slides. Is there a way to see the slides? Or just tell me where we left off, Tom, and I can.
Sure thing. It is slide 6.
Slide 6? Okay. I can just
Neutral
go through it.
convener.
Okay. I'll just say advance. And then, well, thank you. I'm Colin Hopengame. I'm the Vice President for Rare and Orphan Diseases at Critical Path Institute. I am apologetic that I can't be there today. I had a conflicting meeting that we get required to be there, and I would much rather be with you all than be at this meeting. It's a more stuffy meeting. I like working with families and clinicians and whatnot more so. So I lead the rare disease program at CPATH, and I think Tom gave you a little bit of a peek about what CPATH is. What I want to do is continue on and discuss this further, and then give you some examples of things that CPATH has done, but how we engage with communities. I mean, CPATH was obviously founded at some time ago, and we pride ourselves as being a neutral convener. I mean, we truly work through, working and gathering stakeholders. So we bring in, patient communities. Sorry, okay. We work through working with the patient communities. We convene with, regulatory agencies around the world, and we work with, sponsors and clinicians and whatnot, really to really, generate scientific questions and then solutions to advance drug development for specific diseases. And in our case, it's really the, rare diseases that is our primary focus, and particularly rare neurodegenerative diseases is a larger initiative that we have at Critical Path Institute. So the first step we really focus on is really under defining what the unmet need and translating that into what a regulator, such as an FDA or EMA person would try to understand, because, the regulatory agencies are really a bit of the policymakers that really align with deciding what drugs are safe and effective. Once we define the unmet need, we work through and focus on development of basically tools that would, overcome those bottlenecks, such as, biomarkers, clinical outcome assessments, which are, ones relevant to, endpoints within clinical trials. Those are important to proving a drug's, is effective. We have a data management and data science team. We'll talk a little bit about that in a bit. Which is, data is important, particularly in rare disease, because every data point is really precious. We have a modeling and AI group that focuses on, really innovative trial design methodologies so that we use fewer patients for shorter durations to produce the same benefit, but also tools that can help with informing decision making, so that we're making sure that we don't expose patients to trials that would be burdensome or more problematic. And then a regulatory science group. I think one of the things I struggled with when I was a clinician and a researcher is that when I would design experiments, I really didn't understand how a regulator approach the same science. And so it really helped me understand how I needed to design my study better so that whatever I was doing had application to advance drug development or to generate something that could be usable to advance a treatment. And so one of our core areas is in that space. What we do is we're in our role, we are a nonprofit. Once we commit any of our resources, we make them publicly available. So we want to make sure that we de-risk as many areas as we can, but also then disseminate those learnings, those experiences, and those tools, so that those communities can move forward and overcome those bottlenecks. Next slide. Now, the Act for ALS was passed, and it really was a pretty monumental legislation. For us, we were chosen by NIH and FDA to stand up a public-private partnership, which really a public-private partnership is something that generates with various communities, just as our model. And immediately what we did is we reorged as an organization, and we partly did this because we wanted to have shared learnings amongst rare neurodegenerative diseases, because in many instances there's some of the same questions, but they're applied to their unique circumstances. So a lot of institutional memory within the organization that we wanted to capitalize on. We also expanded, and we'll talk a little bit about ALS, but I want to introduce it in a bit of a different context to understand, what we do at CPATH and sort of the steps of a pre-consortium. And then what we've been working on is trying to develop new areas as we move forward so that we can impact better communities. And ones that we've been targeted recently has been, we have an effort in PSP, and one we're trying to forge right now in frontotemporal dementia. But one of the key things, and Tom is an example of why this, our commitment to this, is that within R&D, Tom was the first director of outreach. And what we really wanted to have, within this initiative, of this voice of the community to be even more amplified, more real-time, more frequent and often, because we felt that the keys of success were really imminent there, and that's sort of the goal of this initiative, is to integrate and translate the stakeholders, but the louder voice in the room is we want the patient community. Next slide. Now, what I wanted to explain real quickly, and I mentioned data competencies, and the reason I say it's important is that one of the core, the important parts of our role is that we really work, we have a huge platform that the FDA supported, that would allow us to take in data from all data types. I mean, it's to curate it, to make it, what I mean by that is basically improve it and put it into a common language, which was called a common data model, so that it can be used by stakeholders in sort of the same data language, that are around the world. And we actually have data from around the world. And one of the things that's unique about it is that it's been made also available to regulators. So like if someone say in EMA, they can pull up the platform and they can look at it and work with sponsors to interpret the data. I just had an email before I jumped on this call from CBER, which is the gene therapy parts of the FDA, about working through them about talking about creating some statistical models for trial designs in gene therapy. So they're using this platform as a way to advance drug development along with us. And so, what's nice about this is that it's also shareable to community. When we get data where we openly share it based on the permissions. Whoever owns the original data, we follow those permissions, but we try to incubate that. Next slide. Now, why is this important? I mean, using the example of tools, I just want to give some examples of some tools in this next section and sort of understand what a drug development tool is and why it's important. One of the things that's really challenging when you work in rare diseases is oftentimes there's not been an approved therapy. And there's a lack of a regulator, making decisions about particular aspects of the trial design or how you judge a drug's effectiveness, or even really understanding well the natural history of disease and how disease changes over time. And so one of the things that sponsors have to do is they work with regulators to devise a study, but what's important to that is really not just the duration and the dose and all the other pieces out of it, is the statistical methodology behind the trial. And so how many patients should be involved in the trial, for how long, and, there are subgroups of the study that need to be considered. So one of the things that we commonly do as a drug development tool is create simulation tools. So we take all the data that we have, and we take all the different variables that are listed on that slide, and we synthesize statistical models. We can apply artificial intelligence methodologies, natural language processing, whatever that's needed to really optimize the model. And once that's produced, is that we have that model undergo regulatory endorsement. What that means is that anyone who wants to use that tool, the FDA or EMA will acknowledge that that's acceptable. So if a sponsor, a sponsor does not have to go in, or an investigator doesn't have to go into the Food and Drug Administration and say, you know what, this is my statistical plan, I have to, defend it, everything. It's been endorsed, you've got a green ticket to get into the fast lane. You can immediately use it for your purposes for the study. Next slide. Another, this is just what a pictorial example of what a GUI would look like, or a global interface, is just to give you an example, is you take all of the different variables relevant to that disease state. This is one that's undergoing fit for purpose determination under Duchenne muscular dystrophy. But this is what it would look like. And it's a very user interface. Most people could navigate this, right? It basically uses an interface that you can do to calculate, and you can predict the type of statistics that you want at the end. We've had sponsors use these for various things, even decisions about how long studies, whether or not you should go into a specific space or whatnot, all those type of things. Next slide. One of the things that's often happens in rare diseases is that there's a lack of clinically meaningful endpoints that have regulatory rigor, which means that are accepted by a regulator in a consistent manner. We don't oftentimes understand how much they vary. We don't understand how much variation there is, how they change over time, et cetera. The simulation tool does that. But oftentimes what happens is that when you're working in rare diseases, that there aren't, I'll back up. One of the things that the FDA tries to determine is what concepts are important to patients, what things help you feels, functions, and survives, what things are from that perspective. And so normally what we do is we have measurements that assess those things, right? The survives thing is pretty easy, right? Survival is pretty, black and white, but the other parts really get complicated. And so oftentimes scales that we have are developed for clinical purposes. Those sometimes aren't the same as they're not fit for use for a clinical trial. One of the things that we do as an organization is we begin to, especially if there's frustration with particular clinical outcome assessments, one of the things we do is we actually work on developing new ones. And so, like right now, we're in a process. This is from the guideline from the Food and Drug Administration, but it involves a series of steps that, some of them are literature review, some of them are, interacting with the patient community, but really understanding what are the things that we need to measure and how to measure them. And, then we work with the academic community and NIH, for example, to develop and execute on them so that there are tools that will more adequately capture, the variables that are important to patients. And so and in this instance, we got to get them fit for purpose under the qualification program or the fit for purpose program at the FDA, using that as an example. Another example of drug development tool, one of the problems that happens in various diseases, we don't have the same language. We don't understand how biomarkers change. We don't understand how the clinical symptoms match towards biomarkers or imaging or other things. And so one of the things that we oftentimes do is to create a framework so people can understand the time course of those things, because it helps you identify, where should I put my clinical trial? What most likely benefit am I going to see change? Maybe it's early in the disease, maybe it's later. Really trying to work through what population I really want to study my treatment in. The other part of it is that as sponsors work with patient communities, it's trying to understand how many patients are there using the same terminology. How do we find these people, et cetera? I mean, so that people have, and we've actually created disease staging programs for alpha-synuclein-deletions like Alzheimer's and Huntington's, and there are others that we have active programs in. Next slide. Other one solutions, data standards. Oftentimes when we go into new spaces, there's not a common dictionary that's used for understanding. And so people really aren't comparing apples to apples. And so, we've worked to create data dictionaries that will help the community be able to actually do a better job of taking the data that they have and studying it. We try to make that publicly available. The other pieces that, like right now we're going under we've heard from clinical trial folks and sponsors that there are some diseases where how they do the exams and the assessments for those areas are pretty heterogenic. In fact, even within clinical trials in different parts of the world, they use a different scale, which was a bit surprising. What we try to do is create standards and even ways to analyze the data in those instances so that there's uniformity. Why that's important is that the more variability that you have in an assessment, the more likely that your drug is going to fail in a clinical trial. And we want to minimize that so the probability of success of that treatment and the number of individuals in a clinical trial doesn't have to be as large. We want to try to reduce that. We do biomarker work, largely trying to look at qualifications or letters of support for biomarkers. I mean, I think Tom, alluded to in his section. I mean, CPATH's leaderboard for endorsements is really high, and then biomarkers are one of the key areas. And what that means is that the FDA will accept that biomarker as a specific tool that can be used in a clinical trial versus for all sponsors, not just the one that's particular to a specific drug. We use methodological support. Lots of times right now, we're working with NIH to design natural history studies so that the natural history studies are better aligned with what a regulatory agency could use. So we're not just studying it in a way that gives you a paper, it evolves the paper beyond, and so it can be used for maybe things like historical control groups, so they minimize placebo, or it embeds exploratory endpoints that could be adapted for further biomarker development and things like that, so that we're actually using that natural history study in ways that it was not even anticipated when it was thought to originate. We oftentimes work with different groups to try to advise them on fit for purpose things for data, the data that they have, and also, as I alluded to, things like synthetic control groups in the hopes of reducing the number of patients that get randomized to control arms in clinical studies. Next slide. Now, I want to talk about those are some examples of drug development tools. Tom talked about what CPATH does. What I want to talk about is how we work with communities. And I'm going to come back to that neutral convener role. Whenever I go into a space, and I've come to a lot of spaces in my career, and one of the things that's nice is you're not biased. You come into this world with, pretty much new eyes, and you convene and you work with that community to understand what do they feel is important. What information do you need to advance things? And you take into the different lenses from all of those stakeholders we talked about earlier, and you synthesize that into something that is an action plan and a strategic strategy. And so a pre-consortium model, one of the things that really, starts out is I need to understand where I'm going to go. And I need to design a series of projects that will get me there. And, and whatever that is it can be a lot of options. There's, depending on the disease state and where it's at in its evolution of moving treatments through the clinical trial infrastructure and all the way to approval, it can take on various forms. We come into it with no preconceived expectations expectations of what that's going to look like. But with the ideas are sort of the underlying backbone is that everybody gets a say, and that we want to come up with something that produces, because that's our mission. It's really around, trying to make it to encourage. And I think with areas like this community, one of the things that's key is that we want to de-risk the space. We want to make it attractive. So all these tools are available so sponsors can come and try to explore and begin to work through things. So obviously there's a list there of things that we do as part of that activity. But one of the things that we do work with closely is we get feedback from the regulators who oftentimes, like I said, they're the decision makers, and they're like sort of the buck stops to some degree. One of the things that also we do is that as part of like the pinnacle of it is a research roundtable, that focuses on trying to bring the community together and ultimately a project plan and to launch a consortia that will have multiple work streams that produce whatever tools and solutions that are needed for that space. And then give you an idea of what that would look like and sort of the timeframes that we're talking, within like now. I mean, this is going to be hypothetical as well. The stakeholder engagement would happen over a couple months. Then we'll be beginning to break down all the pieces of that space. So it's basically inventorying everything you possibly can to understand as much as you can about the condition, where all the data lives, what all the trials are, even for ALS, for example, we ran around and we found where all the biospecimens were. I mean, we literally surveyed around the globe what type, iPSC, brain, longitudinal, plasma, can you share, not share? I mean, very comprehensive approach, even talking about things of how to connect the pieces together so that for researchers that are starting to develop this space, they could be able to move quickly towards that. That's not a CPATH thing to go analyze that, but it's to make sure that the community had, this is where we go, this is what we do, this is how you make it happen. And with the idea that we can feed back information on where are the gaps in information that we're seeing. The next part of it is really talking about, creating a more permanent structure. It's fundraising around that structure with that community. It's engaging sponsors. It's, working collaboratively for grants, et cetera, to execute. And those are roughly the timeframes. Typically, consortia will have multiple work streams. They're more complicated and involved. But the corner thing is that the community that engage with them continues on in the process. It's not like you're just one and done and you're off to the curb. It's that community continues to nurture the same initiative forward so that they actually involved in generating the solutions. Now, next slide. This slide really gives you a high-level summary, and you can have these slides. We're good with sharing them because they're all small print. But the pre-consortia for ALS that we just have worked through. I mean, as you can see here, this is actually simplified, but it's these are what six streams. We launched a patient, a PWLE group. We have embedded them in all the work streams. We landscape data. I mean, there are over 70 international data sets with data. We're mapping clinical trials. We're actually working on curating those right now and putting them into regulatory formats and working with the quality for them. Graph data dictionaries, really all with the goal of harmonization. We've already transferred data into the warehouse. We probably are at 3 transfers in right now. We have about 18 other agreements in place to launch that. We have biomarkers, we work with the community to identify the biomarkers that should be prioritized and ones that were particularly most meaningful or interesting to academics and sponsors. And then what we did is we targeted data sets as priorities for ones to go after because we wanted to understand what would be ready for use now, but then what could we feed back to that community, particularly in this case, NIH, of where you need to fund research and where you can fill in the holes so that we can make them usable by the broader community. The clinical outcome assessments, one of the things we've been working on a lot right now is actually, making recommendations to the Food and Drug Administration. We're actually working very closely with the Food and Drug Administration on this project because they hear the frustrations with the primary scale used for ALS, and they've just said, we want you to boil the ocean. And we took that to heart and actually executed a project to look at that. And, I walked you through the method there in that slide. We're actually launching an AI modeling group because we really want to align with the global community in this space. There's a lot of people starting to work in this space. What we want to do is try to encourage the people that are the experts to get in the room with regulators to okay, let's do this. Let's make sure these tools are things that we can move things forward with that they'll accept, right? So you're not, it's not just for that specific, paper or publication, but a regulator is going to take this and say, you know what, I'm good with this. You can use this, a sponsor can use this, we're good with. And then DHTs. One of the things that was very vocal with the patient communities that we interviewed were they really wanted digital health tools because they wanted to have more power over their disease. They really didn't want to travel. They wanted to have, more flexibility in how their condition was being assessed for clinical trials. And so immediately we targeted data we worked with the different researchers to really start understanding how they were studying these things, how their data, and we're working through right now in conversations with the FDA to how to best use this data. And really, what I've told that community is I want an early submission this year, and I want to start the process. I want to get that moving because that was the thing that they told me that was most important to them. So now that's a big beast. That's consortia, would then launch. It would follow up with those work streams. That consortia is fairly large. We have other consortia that have 1 or 2 work streams. And like the ataxia programs, for example, have 1 or 2 streams. ALS is much bigger. A task force, think of it as a single work stream. It has a bit of it's a more of a condensed, it's like a mini consortium. So it focuses on a specific solution that community feels important. And, the idea behind that is to really, focus attention. It produces a single solution. Oftentimes it will involve data. Some of them actually, we're working with rare epilepsy community right now because they're interested in clinical outcome assessments or endpoints that don't necessarily relate to seizures because there are gene therapy studies right now where people are more concerned about neurodevelopment or mobility, things like that you wouldn't have normally thought in some of these progressive epilepsies that could be impacted. And then the idea is that they stand up and they terminate, within a shorter period of time, like a year and a half, and then they're done. They've completed their submission. They've gone through endorsement, submissions and whatnot, and then they're completed. Next slide. Now, this gives you an idea of what that looks like. This is a PSP task force right now we have going on that I alluded to earlier. These are data that we curated, we stored, made it all regulatory compliant, and we're creating a clinical trial simulation tool for that community. I had a call this week and we thought, okay, we're talking about this and we're made progress. We mapped all the data. The academics, researchers told us we're missing data. We'd love to get our hands on it. And so we went after, I'm going after those now. But the other piece of it is we got to talking because there were a lot of people doing disease modeling in this space. And we said, well, we don't want to just stop here. Can we look at biomarkers? And we started talking about MRI studies. And so one of the things that we've started to look at now is okay, let's do this. But then let's blow up this as a side sort of topic in looking at identification of a biomarker. And so we're actually going to be working in that space. And so the next slide, that gives you an idea of how we work through. Is the task force started as really looking at a simulation tool, but it's also going to evolve into something that we'll look after, a biomarker. And, what I'd like to do in that space is that, for those of you who have been in drug development, and there may be some researchers that have discussed this, the qualification program is very complicated. It's very long, it's very protracted, it's very challenging to get through. So what I had advocated for within the group is that, I would hope that we would go for letter of support. And why that is that a letter of support for the regulatory agency means that I can go after accelerated approval with that being supportive evidence, which means I get closer to the finish line faster. And so, we're working with that community to now look at the data and the imaging and the methodologies that they're using so that we can, put that on the list for them to work with us to pursue. Next slide. The next slide is just sort of our contact information. I know I've talked a lot. I hopefully gave enough time for some questions so that if the group wants to address some questions that they might have, I'm happy to do that.
Thank you, Colin. If we can turn the Zoom volume down for questions, then I'll repeat them to him, and then we'll turn it up for him to answer, if that makes sense. So quickly, congratulations, you survived that. It's a lot, and I actually think it's important to understand that it's been a lot of hard work in the community that's gotten to the point where you can sit in a room and have a pretty dense scientific discussion about the drug development pipeline. That's exciting. So congratulations that we've gotten there. Now for questions, I'm sure that there are a lot, or there may just be like glazed looks. Don't worry, we'll make the deck available. I'm going to be around, so if, as you get a chance to digest a little bit, feel free to come and find me. Also, our contact information is up there, and I sincerely welcome any questions or outreach later on. Excellent. Oh, we have a question.
Here's our first question.
Thank you for the presentation, and all the work that you guys are doing seems very important. The question that I have is, what does the intake process look like? How do you identify, say, a specific thing that you want to take on and connect all of these different pieces together?
Great question. Colin, did you hear that? So the question was, what does the intake process look like, and how do we go about identifying targets for gathering information?
Sorry, the intake, could you please clarify the intake process for which part? I guess that would be helpful. Is it for data? Is it for our pre-consortia or task force, or which part is?
I was thinking more in terms along the lines of taking on a new project, like taking on a new, say, if we're looking at HSP, a specific mutation, and connecting all of the different pieces together with the research that's out there, the patients, the biomarkers, and the whole end-to-end system.
So disease states.
Disease states, it really depends on the communities that we work with. I mean, we try to find out, where there's sufficient, interest and data and stakeholders that are willing to work together. And that's the biggest part. I mean, the only other piece is really important is just our sustainability piece. Like for initiatives under R&D, part of the funds would most likely come through that act, but we would also, because it's a PPP, the community. The other part of this is that what I've been working on, and I think I mentioned this to the board, I know the program officer for HSP and SP, and we're talking, and one of the things that we were hoping was to draw attention more to this space. He's oversees the portfolio, and I told him that I was giving this presentation. He said, well, I was going to talk to our board, our oversight about HSP and SP, and try to see about convening about some of the same issues. And so so there's those opportunities also as well, and how those all kind of connect. We learned a lot with ALS under this, because it was just this bigger strategy between all these different governmental agencies and whatnot. What I told him is that my hope would be is can we do something that's at least even a smaller version of ALS, because ALS has got a lot of stakeholders, and try to maximize that. But honestly, it's the community that really guides us there. I mean, I'm across rare diseases. We've got a lot of different diseases we're working in. To me, it's still not enough, because there's a ton of rare diseases. What we're trying to do is be as impactful as we can, and where we feel that the community will work with us to move things forward. As you can imagine, there are communities that, and this may be offensive. I'm a small town kid from Iowa, so I'll say it. But they're siloed. There's a lot of it's hard where people won't share, or they're and those are spaces where I'm very vocal usually about it, and I'll say it's like we can't move our mission forward if we can't work together. And so those are usually a no's for me, and it's because they don't want to share, they don't want to collaborate, and that's unfortunate.
Okay, we've got one more question for you before
To
we
give you
go.
more practical steps is that we sit down with those communities and we define what they need, and we fill in the pieces that, and we work with them. So like, for example, when we do, I'll usually, Tom, you can chime in here too, like with ALS. We have like every other week meetings with ALS community. We have a lot of feedback about how things are going, where things are going, and so it's not like, you just talk to somebody and then they just go off and do stuff. It's longitudinal and interactive.
One more question.
So with
Okay,
CPATH,
what's the question, Tom?
with CPATH, I was curious if there's any red tape or anything that might slow down existing processes that we have in place, like with our CERN.
I think eventually we're going to figure this out. So the question was about red tape with CPATH, and if our process would slow down what the foundation is doing.
I guess I'd have to think about what that really means. So, like, to me, I was vocal about it a bit yesterday. It's really getting to an executable plan and with clear objectives. CPATH is not a huge nonprofit. It's not like, we probably have about 170 employees. We're not an NIH or FDA or, a pharma company or, a March of Dimes or, whatever. We're pretty aligned. Usually the things that slow us down are the same things that slow everybody down. It's the contracting process with different individuals. I can tell you we're pretty liberal on things because we still act like a smaller nonprofit. I don't think that's the issue, what I would say. In fact, that would frustrate me to no end. This isn't recorded, but that's been one of my frustrations with ALS, is that we're sandwiched between two large regulatory agencies and government agencies. It's been slower than I would have liked. That's why you see like all those working streams literally took us probably, we knew where we were going probably within 6 months. The data stuff right now, it's not us, it's the trying to get through institutions right now. And Tom can attest to this. The reason I threw out that many DCAs agreements to get data in was because I want to get moving. And but since the prioritization and strategic, I've been very strategic about trying to get things that will get me quicker wins fast as possible that address the needs that were vocalized to us. So I don't think that is, and back to the accountability piece. Part of the reasons I'm very passionate about getting on calls with the community is that I need you to keep me in line. If we're not meeting what you all need, it's important to hear that, because our mission is really about producing things that will benefit the community. CPATH started as the FDA said, well, we can't work on this stuff, but we need an entity that can. And so that's hence our relationship with them. And so the bureaucracy for me most likely ends up is that you're working with same things as sponsors will have to work with, or same thing as an academic researcher filing an IND would have to work with. You know, those types of things.
Awesome. We're at time, but
Don't worry, the answer will be 15 minutes. So you can talk with Tom a little bit right afterwards, and he'll give you all the answers.
know how to become a guinea pig.
You're on
That'll
mute.
take-
I can't hear.
I'll talk to you out in the lobby about that one. Okay.
awesome. Thank you, Colin.
Thank you.
Thanks, guys.
Everyone.
Thank you for having us. And if you have questions offline, please feel free to engage. We're happy to meet offline or do whatever you all need, okay?
Perfect.
Okay.
Thank you, sir.
Take care.
Thanks, Tom. Okay, real quick, once again, dinner in here tonight, 6:30. Little cash bar in the lobby at 6 o'clock, okay? No matter what the program says, this is what it really is, okay? Got that? Just one more time for that. Okay, we're rolling right along because we're moving really good. We're right on time. Next is Natalie Downs with Rare Base, from Rare Base. And she's coming, we're setting up her presentation as she's easily sliding up here to the stage. So she's gonna come on up and we're gonna get her going. Look at that, it's almost ready. There you go.
How do I operate this?
I don't have any idea. I think you hit the either the go button or the laser. You can laser somebody, or you hit the right button probably to advance, I would assume.
Okay.
So, or if not, you just tell the man.
Okay, I will.
Tell Harper, he'll take care of you. All righty, guys, here we go.
Can you hear me? There we go. Hi. Welcome to the Drug Discovery Hour. It seems like this is such great timing and a great talk to follow. What I'm going to be talking about is a little bit upstream of the regulatory drug discovery and drug pipeline that we just heard all about. My name is Natalie Downs. I am a genetic counselor by training, and I work as a senior project manager at Transcripta Bio. It is formerly known as Rare Base. It's on my tag. It's in your program, but we changed the name about maybe 6 to 8 weeks ago because we started really focusing in the rare disease space, but we've since then scaled our platform, and now we're able to really interrogate all genetically defined diseases based on the transcriptome, which I'm going to talk a little bit about in our next slide. So I'm very grateful to be here to speak with you all. I'm also here a Tampa local, and I wanted to welcome everybody to the Tampa Bay. Hope you're enjoying it. So let's talk about the transcriptome and what we do at Transcripta Bio. The real foundation of biology is called the central dogma. It's the idea that your genes are held in your DNA. And DNA and genes are read by creating an intermediary step called the messenger RNA. And it's from the messenger RNA that your cells read and translate it into protein. So the genes to RNA to protein is the central dogma. And your genes make up your genome, but your RNA, as it's being expressed, makes up your transcriptome. And that is highly variable. It varies by cell, it varies by what the cell needs in that moment, day to day, minute to minute. And that is translated into protein, which provide all the functions the cell needs to do its jobs. So we are at Transcripta, really instead of looking at the DNA like you would do in a genetic test at your doctor's office, we look at the next step over, the RNA, and evaluate what genes are being transcribed in that cell in that moment to try and look at the different patterns of gene expression. So we do that through a tool called RNA-seq. And RNA-seq, what makes us a little bit different at Transcripta Bio is we perform this robust RNA-seq on the whole transcriptome in human-derived cells. We've started in human-derived neurons made from human stem cells, iPSCs, and we can culture these human neurons in a dish, extract their RNA, synthesize it and sequence it, so read what messenger RNA is being transcribed in those neurons in that moment, and then look at comparative levels of gene expression. We use those comparative levels of gene expression, oh, there's my clicks, to try and look for opportunities to counterbalance any sort of genetic changes that drive disease. So for example, in conditions, autosomal dominant inherited conditions, where there may not be enough functional protein, one copy of the gene is mutated, we can look for opportunities to try and drive and increase the gene expression of the wild type copy. The converse is also possible, that we can look for opportunities in genetic disorders caused by a gain-of-function mutation to try and decrease the expression of that gene in that cell type of interest. For other situations where there may be a complete loss of function in that gene or a missense change that alters the protein function, you wouldn't really want to mess with increasing or decreasing its expression, we can look for alternative ways to kind of work around that gene. So looking at the whole transcriptome to evaluate compounds or methods to change the pathway that that gene is in to try to mitigate the effects of that gene. We do this using our Transcriptobio Function-Seq platform, which is what we call our RNA-seq pipeline, where we, like I mentioned, can use RNA-seq to evaluate the whole transcriptome, the expression of 20,000 plus genes at once in that cell. And that includes the 3,900 or so genes that are currently known to be the root cause for genetic Mendelian disorders. We do high throughput drug screens where we put thousands of drugs, one at a time, but all at the same time in an array, on these cell models, these human-derived neurons, and we look to see how those small molecule drugs are changing gene expression. We can take that change in gene expression using the transcriptome and look at what drugs are doing to dial different genes up and what drugs are dialing certain genes down, and evaluate those patterns or genes of interest. If there's one gene we can look for, what drugs show an efficacious response in the direction you'd want to go to mitigate or counterbalance that genetic disease. We've been using this massive amounts of data that we've generated. So we started back in 2020, I believe, and we've been amassing tons and tons of data through these high throughput drug screens. Like I said, in glutamatergic neurons is where we started, in order to amass a ton of data and look for repurposing drug opportunities, where we're looking for FDA-approved drugs that we could use to repurpose. They're approved for other indications, but maybe we find they have a beneficial effect on a gene of interest, and could that be repositioned or repurposed for that condition instead? So using all of that data that we've amassed in this effort, we're also able to now incorporate AI and machine learning models to learn what small molecule structures are doing to gene expression. And using AI, we can start making predictions about what new molecules could be designed to derive the same or desired effects on gene expression. Those new molecule structures can then be synthesized and put back into our wet lab in Palo Alto, California, and screened in the cells again and close the feedback loop. So we're able to continually iterate on our AI pipeline using real-world massive amounts of genetic expression data. We call this our conductor AI. So like I mentioned briefly, we've started over the last handful of years in glutamatergic neurons. These are in the central nervous system. There's many different neuron types in your brain. This is just one of them. We started with glutamatergic neurons over the last few years because when we started with just a singular rare disease focus, the majority of rare diseases are, especially in the pediatric space, are neurodevelopmental. So we wanted to start in the CNS space because that had the biggest population of potential patients to get going. In glutamatergic neurons, we screened over 3,800 compounds at a single concentration. These included both FDA-approved drugs and experimental drugs that had not yet been approved. And then from those top hits in our single concentration screen, We selected about 400 compounds to validate further in our neurons, where we tested those 400-ish compounds at 10 different concentrations and looked to see if they had a reproducible effect in what we call a drug-like response. With increasing drug concentration, are we also seeing an increase in gene expression like we would expect to see? So we've had great results there. We're very happy. And then from there, we decided the next thing we should really try is motor neurons. So in the last couple of years, we've been onboarding motor neurons, getting that assay set up for a pilot test of about 70 FDA-approved drugs, as well as experimental compounds, to try and understand how transferable our findings from glutamatergic neurons were to motor neurons, as well as get those assays ready to go, with the hopes that one of our next big screens would be testing hundreds or thousands more FDA-approved and experimental compounds in motor neurons to look for new hits. As I mentioned in the last slide, in addition to our motor neuron pilot, we've also been developing AI-enabled screening, so taking the lessons and the data that we've learned so far and training our AI models with it, and using that to predict new structures and new small molecules that we've then put back into the screening pipeline. We've done one screen so far in glutamatergic neurons using brand new molecules we had synthesized based on AI predictions. And so far, that has had very promising results. We're very happy. And we are just gearing up to do our second round of screening with AI predicted molecules, hopefully in the next few weeks. And then coming up next for us, we would really like to move into new cell lines and expand our screening and expand our data into new cell types, new disease types. We're exploring cardiology, ophthalmology, dermatology, and ideally expanding our screen that we've already piloted in motor neurons. So far in the last 2 years since completing the screen and really digging through the data, we've had a number of different successes that we're very happy about. It's early, but we've got about 5 programs so far where our partners, working with our partners, we've identified FDA-approved compounds that are repositioning opportunities. So these compounds were already approved for different indications, but showed some potential efficacy in our model neurons for the conditions listed here. And in all of these 5 cases, these compounds are being tried in an N-of-1 trial or an off-label prescription in a patient. So they're in the clinic getting tried right now. We've got very promising results. We're very optimistic for a number of them. And then down below, you'll see a number of late-stage asset opportunities that are at an earlier stage in discovery, where these are the experimental compounds that we've also been evaluating. They're not yet FDA approved. You can't go to your doctor and get a prescription for these. But they've been through the FDA approval pipeline and maybe past phase 1 or phase 2 for safety in humans, but it failed out due to efficacy in phase 2 or phase 3. They weren't effective for the condition they were tested for. So can we use our transcriptomic platform to test those compounds that are safe but weren't effective and find new things they might actually be effective for? And that's really something we're exploring and would probably fit very well into the drug development pipeline that was just discussed in the last speaker's presentation. Like I mentioned, the next steps for us really ideally would be branching into new cell models. We're very interested in the neuromuscular space and the unmet needs there. We've got the motor neuron pilot that went very well, and we'd like to expand on it. We've onboarded cardiomyocytes, retinal pigment epithelial cells for ophthalmology, and keratinocytes for dermatologic genetic disorders, with the idea that the more therapeutic areas we can build into, the richer our data set can be to find new repositioning opportunities, new drugs that we could find new indications for, but also to develop novel drugs and novel molecules to move into the drug discovery pipeline as efficiently and safely as possible. Now, I took the liberty of looking at our motor neuron pilot data for some HSP genes. I know there's, over 80 HSP genes and many might be missing. This is just a top selection specifically of mostly autosomal dominantly inherited ones. Autosomal dominantly inherited conditions work very well for our platform because, well, one allele may be mutated, the other is usually a wild type, and we can target that for increasing expression of that gene to maybe try and mitigate the effect of having a decrease or a haploinsufficiency in the other allele. So on the rightmost column, you'll see that it's ordered by the expression of that gene in our motor neuron data set. In the unit here is TPM, which is transcripts per million. What our cutoff is to be able to really measure the RNA transcripts in our cells is 10 or above. So anything under 10 is just, it's too small to be detected, or more importantly, to detect subtle changes in expression for those genes. But anything 10 or above kind of meets our threshold. And then, as I mentioned, the autosomal dominant conditions are ideal for us, but I've highlighted here that, for example, SPG7, I know is common. It's an autosomal recessive condition, so both copies of the SPG7 gene will have a variant or mutation in it in order to have that condition. So that might be a target for the alternative pathways I discussed at the beginning, where we can look for a paralog or a gene that's similar that we could try to upregulate, or we could look at the pathway and see, can you upregulate something else and kind of work around and mitigate the effects of that mutation. Lastly, I have bolded some of the genes that just in our pilot study of about 70 compounds in motor neurons, we have at least one candidate compound that seems to upregulate these genes. So I'm hopeful, very optimistic, that if we test many, many, many more compounds, FDA approved or otherwise, in motor neurons specifically, we'll find even more candidates, hopefully for all of the hereditary spastic paraplegia genes. Now this kind of falls in line with a proposal that's currently in hand with the SPF board. They've been fantastic to get to know over the last 6 months or so, where we've been discussing how could we partner to get something like this off the ground and really get some more robust small molecule drug discovery data from motor neurons and motor neuron screen. And in a successful partnership, we really look for opportunities to collaborate with experts. We have scientific advisory boards. I know SPF does as well. And the more that we can collaborate with the experts, even in this room, the better, to share ideas. We could the plan would be to form a joint steering committee to evaluate the data as it comes in at each milestone and decide which hits and which genes to move forward that would be optimal for the community. It's important to have robust data sharing between the two, and anything that comes out of these findings we would hope would be opportunities for joint development, whether that means taking FDA-approved compounds into N-of-1 small clinical trials or off-label prescriptions with clinical partners, or unapproved late-stage assets that could be moved back into the FDA-approved development pipeline to get approval and get it into the clinic efficiently and safely. This is a little breakdown so far of the partnership proposal that we've shared with SPF. Like I mentioned, it would be very similar to our glutamatergic neuron screening, where we start by screening about 1,500 FDA-approved compounds in a single concentration in motor neurons, followed by selection with the Joint Steering Committee about what the top candidates, the best data, top hits, top genes would be based on that single screen. We can then move those into CRC screening, which is the concentration response curve. It's an industry standard assay to look for drug-like effect, where you test the drug at serially increasing concentrations and measure gene expression and how it responds. Using that, the next step and kind of final step would be testing the best hits for further validation studies. There's a little bit of room here, but it could be something like doing protein analysis. Does increasing gene expression translate to increased protein? Do we see functional effects on imaging or any other assays to measure how motor neurons are functioning related to that exposure to that small molecule? So this would be the proposal in hand. We're very excited to hopefully continue talking to your board, your team, and learning more about everything that you get to do. We hope to be a part of it. And with that, I thank you all very much for your attention, and I'm happy to take any questions.
Okay, I have a question back here.
Hi, I'm assuming that you guys have a proprietary AI. How are you preventing from hallucinations? What are you doing to mitigate that?
That's a very astute question. Yes, we've been designing our AI algorithms in-house. We've got a fantastic team working on it, including a whole battery of chemoinformatics folks. And we're doing this development based on our own real-world data. So we've spent since 2020, doing lab work and testing these drugs on cells in-house, in the lab. And so we're just using our own proprietary data to train those algorithms. Those algorithms can then make predictions. And here's where we close the loop. We're able to put them back into our wet lab and test them again and see if they were right. So you don't really have to just trust it. We get to actually put them back on neurons in a dish and see if they were correct.
Any other questions? Oh, okay, I got one more right over here.
Thank you. Great presentation, thank you. I need help understanding something, though. Sure. Going back a couple of slides, TPM, tell me the significance of the number.
Sure, no problem. TPM stands for transcripts per million. And I have a hard time with it too. My background's in genetics, and RNA is kind of a new area for me. But RNA is a variable thing. It changes depending on the needs of the cell at that time in response to its environment. It's just measuring how much that specific gene is being expressed. And we do that by trying to basically pull out the transcripts for that gene. So trying to kind of segregate the RNA for that specific gene and quantify it. TPM is one measure, but there's also other ways to use normalized measures. It's just the one that we use because we can set a nice cutoff of can we detect that gene at a robust enough level so that if there is any changes to it, that those are statistically significant and able to be detected. If the gene's like barely expressed and then you make a small change, you can't tell if it's just by chance, fluke, or is it a statistically significant difference?
Hey, Natalie, we got a question over here.
This was an excellent presentation. I missed maybe a little bit in at the very beginning. So, the repurposing, finding the drugs that works on motor neurons is very important. And, there are many FDA-approved drugs out there, also new ones. So for your drug screening in motor neurons, so can you remind me again, how did you put those motor neurons in? Is it in culture? Is it in silico? Is it in vivo? What models do you use?
Yes, good question. So we did a very similar process to the glutamatergic neuron process. I believe that we started with human stem cells, and we can drive the human stem cells. They're usually, pre-differentiated to neurons, and we can drive human stem cells to become specifically glutamatergic neurons, or in this case, we were also able to drive them to become motor neurons.
Spinal motor neurons. Yeah, but this is the disease of the upper motor neurons. So that's usually the problem with the iPSC cells or, derived motor neurons. Then, first you have programmed cells, like they are engineered, these are pretty much made up cells. And now they're also not upper motor neurons. The only thing is that the species is human.
Yes.
But you may as well work with HeLa cells or other cells. So the glutamatergic neurons, you make them the brain glutamatergic neurons, right?
Yes.
But they may not really be the neurons that you want to study because they may not be the bed cells, they may not be the corticospinal motor neurons, correct?
That's a good question for our cellular scientists. I'm not sure on the specifics, but that I do know that they're human-derived iPSC, glutamatergic, and motor neurons.
Okay, all right, thank you.
Yeah, thanks.
Okay, we got time for one more. All right, well, get ready now because y'all are going to be stretching in a couple of seconds. Natalie, great job. Thank you very much. Okay, guys, you asked for it. Time to take a little break. We got mini stretching with Zachary Lipson. Here we go. Here he is. Here he comes. Welcome. You can do it from the podium, or you want this?
I'll take this.
Okay, good.
Good afternoon, everybody. How are we all doing? I know we've been sitting down. It's been a lot of science talk today, so we'll be taking a little break from that to bring you guys some stretching as well. As you guys can see, I almost just tripped. I have HSP as well. I have SPG4. I was born with SPG, or with hereditary spastic paraplegia. Understand some of us here might have developed HSP in our lifetimes outside of adulthood, but either way, I am here to help you. Consider me as your guide. So without further ado, let's get started. We've been doing a lot of sitting today, so everybody, let's just start with one big good morning stretch. Bringing our arms all the way up. If I talk like this, can you guys hear me? Yes. Okay. Let's just take a couple deep breaths. Breathing in through your nose, out through your mouth, stretch it as high up as we can. Okay, you guys can bring your arms back down, and we're going to do some moves that a lot of you may hear people who do yoga are doing these, obviously not sitting down, but everything we do today can be done sitting down. If you'd like to sit up, please feel free. So we're going to start with a little bit of cat-cow next. So if you guys would like to put, is this on? Hello? Okay. So if you guys would like to put your hands on your knees, if you're sitting down or standing up too, we're going to be rounding our backs. So as you round your back, let's take a deep inhale. And then we are going to bring our back the other way, bring our stomach forward, and take a deep exhale. And this is called cat-cow. So as you inhale, we're going to round our back, and as you exhale, let's flatten our back out. I want you guys to do a couple on your own inhales and exhales, sitting or standing. Then we'll come back together in a couple breaths. Okay, everybody, let's return back. And from here, just a classic stretch. We're going to reach our left arm all the way across to the right side of our body. And let's switch sides right here. And we're going to go one more time through. So let's bring our left arm back across. And one more time with the right. Perfect. Great work, everybody. Okay, so from here, we're going to bring our arms all the way up around to the top as we take a deep inhale. And just exhale our arms straight down through the bottom. Let's inhale, going back up all the way around. And when we bring our arms down, you can follow along with me. We're just going to go straight down. You can bend forward in your chair. If you're standing with me, you can just bend straight forward. And from here, everybody, let's start to make our way back up. And we're going to bring our arms all the way back up to the sky. Beautiful. And one more time, let's exhale, bring our arms back down, and then we will finish again with our bodies. Okay, good job, everybody. Let's return back. We've been moving our bodies. We're going to get our arms moving a little bit. So if anybody has ever tried boxing before, we're just going to do a little bit of hand movements. So everybody, let's take our dominant hand. For me, my dominant hand is my right. We're going to consider our right hand. Sorry, I'm messing this up. whichever hand is not dominant, we're going to consider 1. So for me, my right hand is dominant, so my 1 is going to be my left. And then when I say 2, that'll be our right hand. So we're just going to throw a classic. We're going to move our arms a little bit. 1 and 2. You can do this sitting down. You can do it standing, whatever you'd like to do. We're just going to get our arms moving a little bit. Nice work, everybody. Nice. Hey, I'm seeing a lot of smiles today. Okay, so now we're going to incorporate some more. So that was 1 and 2. Now we're going to do 3 and 4. So when I say 3, you're going to bring the hand opposite dominant around the side to the front like that. Perfect. And then when I say 4, we'll do the other hand. So now that's going to be my right, and I'm just going to hook. Bring my arm straight out to the side. Perfect. So now let's just do it all together. 1, 2, 3, and 4. So it will look like this. Simple, right? Okay. Now we're going to throw all of those together. We're going to do it all together. 1, 2, 3, and 4. We're going to do it a couple times. It might look funny, but it doesn't matter. Nice work, everybody. Okay, and we have 2 more to add. After this, these are the last 2 we're adding. So our 5 will be the hand, number 5 will be our hand opposite from our dominant again. And from here, this is called an uppercut. So we're just going to bring our arm straight up to the sky. Yep, and then classic, 6 will be the other arm, just like that. Nice. So let's do them all together. 1, 2, 3, 4, 5, and 6. Just like so. Nice work, everybody. Wow, I'm seeing a lot of strong punchers out here. I don't want to mess with a lot of you guys. Just keep throwing them for a little bit longer. Okay, great work, everybody. Let's just shake out our hands. Okay. So let's bring our arms straight out to the side. And however far back you can bring your arms with our thumbs up, just slowly try to retract your shoulders. And if you can't retract them, if you're having trouble bringing them back, just keep them straight up. And let's bring them all the way out to the front, keeping them as extended as you can. And let's bring our arms all the way back out once more. And bring our arms in. Okay, so for those of us who can, we are going to just shake our legs, and if we can't shake our legs, we have our arms, just shake our arms. Okay. So we did this in the beginning, but we're going to do it one more time. This is called high mountain, so I'm just going to call it what it actually is. So we're going to inhale into our high mountain pose, which is bringing our arms straight up. Then we're going to exhale, just bring our arms all the way down. Let's inhale, bringing our arms all the way back up to the sky. And exhale all the way down, bringing our arms down to the ground and letting go of our back and top half if you can. And let's inhale one more time into high mountain pose, bringing our arms all the way up to the sky. And release. How are we all feeling so far? Good, good, good. How are we looking on time? Okay, we got 5 minutes. We're going to end off with some more stretches. Okay. Let's bring our arms straight out. guys in the back can hear me right now? Okay, cool, just making sure. Bring our thumb straight up. Retract those shoulders as far as you can. How many people in here have swam before? Okay, for those of you who know what streamline is, we're going to bring our arms up into streamline, but if you don't, that's just where we're going to bring our arms straight up to our head and try, if you can, keep your arms behind your ears. If not, just straight up like this is fine. Let's retract back down to the ground. And one more time, and in streamline. Okay, everybody, can retract back down. And I hope you all are feeling good. I know we've been doing a lot of sitting today, but I hope that woke you up. I saw a lot of smiles. So I hope you all had a great stretch. Thank you very much.
Okay, everybody. You guys did real good. Really proud of you. You did good. You got some stretching. There's going to be another opportunity to stretch with Zachary after our next couple of presenters, okay? So you're going to have to do this one more time today, okay? A couple of quick notes while we've got a couple of minutes. When we do our breakout sessions later at 4:30, we're going to make sure we want the young adults group who want to get together. We're going to have them kind of meet in the lobby, or they can meet with Blair at the registration table, okay, for that breakout session for the young adults, okay? Hey, I got another note. Tomorrow at 5:15, Saturday at 5:15, we're going to be taking a picture with everybody who's got their 5K t-shirts on, okay, from all the 5Ks you've done over the years, okay? Wear one tomorrow, and at 5:15 we're going to get a group photo, okay? Because it's 5K season now. It's been open from March and going through October. So you got plenty of opportunities to do a fundraising 5K. So if you got a 5K shirt, wear it tomorrow, and we'll take a group picture probably out here in the courtyard area outside of the ballrooms. Okay, so that'll be 5:15 tomorrow. Not tonight, tomorrow, okay? So outside of that, we're moving right along. Things are going really, really good. We're getting ready to do our next presentation, and I'm waiting for him. There he is. Harper's in place. Ladies and gentlemen, we're bringing in, we're making a flip now on your program again. This time we're going to start with Dr. Craig Blackstone. Here he comes, right now, coming up now. We flipped over. A little change on you. Keep you guessing. Here he comes. have this tool here. There you go. I think you just hit the right side, I believe, and it should advance. And you kind of have to aim it at the laptop back there in the right back corner with Blair.
Okay, sure.
Okay,
Thank you.
There you go. You're good to go.
Thanks. Okay. Well, it's a pleasure to be here, everyone. As many of you know, I've given presentations at these for many years. I think something like 15 years ago is when I first started coming to these meetings, and I know that many people are new. Oh, it's not on? Oh, sorry. Oh, sorry. Is this better up here? Okay, yeah, I think it was a little too low for me. Sorry about that. So I was just kind of saying that this is, I think, I've been here about 15 years or so to these meetings, and it's always a sort of a highlight of the year to be able to meet with all of you because, as you know, I've really focused on this disease ever since I first became a faculty member well over 20 years ago. And it's really remarkable to see the advances that have taken place in the HSP field to the point where I used to talk about basic science and a lot of the things that we were working on in the lab. And now we're really talking about therapies, you know, how we're going to bring all these great insights that have been made over the past couple decades, both genetics, basic biology, model systems, how we ultimately bring that to the care of people like you that have this disorder. So it's really an exciting time. When I was putting my intro slide together here, just my title slide, you know, I thought one of the things I noticed about this meeting and about HSP in general that you might not all realize is that many of the researchers, physicians, and so on that give talks here and work with HSP are from very different backgrounds. So you'll see I'm a movement disorders doctor. So the other kind of patients I see are Parkinson's, Huntington's, tremors, dystonia, things like that. Whereas Dr. Mitsumoto will come and he's primarily a neuromuscular doctor, sees HSP, but also ALS and other motor neuron diseases. And I know other people you've heard from, like Darius, have a training in pediatrics. So I think you'll see that a lot of us really come from different backgrounds to the HSP field. And I think that has the advantage of us all bringing the insights that we get from those other diseases that we study, to benefit people with HSP. Another thing I do is I oversee research for a very large department, and that has also given me a lot of more general perspective in terms of what's out there in terms of new therapeutic development, so we can take advantage of all the advances in stroke and epilepsy, brain tumors, Alzheimer's, Parkinson's, and all that, and see how what we can learn from those disorders that might benefit people with HSP. So I will get on to my first slide. So I do have some disclosures, and again, these are companies that we often work for where we get our money. One advantage, even though I won't talk about anything related to companies today, is that it does give us the perspective of how to work with industry, how to work with biotech, and how to work with pharma. And many of us who train in basic science or in clinical medicine in the academic track really don't understand the business of medicine. And it's been very valuable for me to understand that because there are partners in bringing therapies to people like you. And to the extent that we get to work with them, I think has been very valuable for me. And I might share one or two general insights about what I think industry is looking for from people like us to be able to move therapies forward. Now, one of the, also I think, areas of diversity among all the different people that speak to you and that you hear from is that our institutions, though they're all academic, are very different. Some of them are small, some of them are large. They're in big cities, small towns. And I think that that's also a tremendous strength, that we all have different perspectives. So I was going to just spend one slide just talking about what I think my institution can bring that's relatively unique. And here's a picture of part of our hospital. So it's very large. We're the largest neurology department in the United States, and we're merging with another hospital where we'll be even much, much larger. And that has disadvantages. I haven't met many of the faculty that I work with ever in 3 or 4 years. But at the same time, we cover just about every disease, just about every type of trial is going on, and we can learn from one another tremendously. And this is just a part of our hospital. All those buildings are part of Mass General, and then we have a whole other section for research buildings and several $1 billion of new buildings on the bottom of the screen that are being built. The advantage of that is that we do have the ability to learn from lots of different areas of medicine. You'll see a bridge at the top there, and that bridge goes to Cambridge, Massachusetts, and that has the largest collection of biotech firms in the nation, and probably the world. So we interact very closely with them, and we're hoping that that enables us to bring therapies to people like you much quicker. Also across the river, and this is all walking distance, is MIT. And MIT is an area, a university that we work with very extensively on new technologies, both in terms of research as well as in neuromodulation and assessments for clinical trials. And I'll mention some of those as well, because even though we're often using those in other diseases that are far more common, all these insights will ultimately get to benefit patients with HSP. So one of the things about therapy that I wanted to emphasize first is that a lot of the therapy that you get now is based on circuitry. So we understand, and I think you've probably seen this many times, that these upper motor neurons are involved in both PLS and HSP. And of course, one of the differences is that HSP has all these genes that you get identified, though, as you've heard many times, a lot of people with HSP don't have a genetic diagnosis. And that's why there's so much emphasis on trying to, find additional genes, find additional types of mutations, to be able to give people an answer and move closer to therapies. PLS, of course, doesn't have as much of the length dependency, though it does have some. And of course, a lot of other upper motor neurons that are above the waist can be involved to a greater degree. Of course, there's overlap between these two in the cells that are involved, and we're hoping in terms of the basic mechanism so that a lot of the benefits that we see in research from HSP patients will hopefully benefit those with PLS and vice versa. Again, one of the, I think, as somebody who oversees a lot of different types of research, one of the really unique features of HSP is the number of genes that we have. And that can seem daunting at times, 90 or 100 genes, you have one of those 100, how are they going to treat this one disease? But as many of you know, we started to think about it in terms of different groupings. It's not 100 different diseases. There's a smaller number of them within various categories. And I'll show more on that later. But in terms of how we think about the circuitry, so you have these upper motor neurons that start in your cortex and go down to your spinal cord, and they synapse onto these lower motor neurons at different levels, and that controls our movement. A lot of our therapies, and this is not an exhaustive list, but I think there's something you'll notice. Obviously, number one is a very effective one, and that's what you were just doing. Stretching, physical activity is very important. But we also want to decrease the spasticity, relieve pain, treat other types of symptoms that can be associated with HSP. And again, there's just a list of different things. But what you'll notice is that in all cases, they don't affect the progression of the disease. They can alter how you feel and certainly can lead to improvement in your quality of life, but they're not what we're really looking for. We need to have therapies that stop it from progressing or even restore some of the function that's lost, or ideally prevent it in the first place. And we do these kind of meetings and trial designs for several other disorders like Parkinson's. So we're always talking about this for other diseases too. It's not unique to HSP, but I think it's important that we keep our eye on what that goal of really trying to prevent or prevent it completely, or restore function, or prevent progression, that all of those things are going to be targeted at cells rather than circuits. So if you think of circuits as the nerves synapse on each other and ultimately lead to movement, your body's made up of individual cells, and all these genes, of course, function within those cells. So we want to drill down to that cellular level, and that's where a lot of the therapies that you hear about are based. So this is an old picture, but I think in many ways, it's still how I think about it and how many people might think about it, is that we have all these genes and they sort of go into different categories. And those different categories means that these diseases might be similar, more clinically as well as in terms of basic mechanism. And our hope is that as we treat any of them within these categories, that other patients with different forms of HSP that fall in that same bins would benefit as well. And I'll show a specific example of that from our own work, but I think it is important to think about that, we have all these different genes and it seems daunting, but they really do seem to coalesce around a couple of common themes. And those themes are areas, of course, for therapeutic development. Now, one thing also, as you hear many times, is that even though it's the corticospinal tracts that are involved in this disease, that doesn't mean that's where the primary problem is. In many disorders, the problem can be in other cells in the brain that interact with those neurons. Like in the case of these, you'll see this up on the upper right-hand corner, you'll see oligodendrocytes, things like that. If those cells are affected and the myelin is disrupted, then the axons underneath that can be disrupted. So even though we think of it a disease of motor neurons, And in many cases, that's probably where the problem starts. It's not the only possibility. The other possibility is that it starts in other cells and that it impacts those neurons. And all those are important things for us to understand in terms of therapy. But I think that over the last couple decades, this idea of thinking about the HSPs in different subgroups, I think is very valuable scientifically, and I also think it's going to be very valuable as we ultimately move toward therapies. Now, this is one example, and I'll just zip through a couple of these just to throw them all up there. So you can see there's examples where we pick one part of a cell. In this case, it's the endoplasmic reticulum. So it's a very big organelle because if you have a corticospinal neuron that's 1 meter long, the organelle is that long too. So it actually goes the entire way down the axon. And if you disrupt that in any way, you disrupt the axons and you get symptoms of HSP. And we get that in a lot of the common forms of HSP as well as some rare ones. So if we imagine that spastin, elastin, and some of the really common HSP genes are involved in this process, we could have an impact on a very high percentage of people with HSP if we could unravel what the problem is here and come up with a way to treat it or mitigate it. So again, this is not a new idea. As you can see, the article that this is from 12 years ago. But in many ways, I think we still think about it this way, that this is an important pathway, and it's one of the primary ones that we focus on in terms of developing therapeutics for HSP. Oops, there was one more. So what I wanted to do is, before I launch into one specific example of how we can use different types of therapy for even a very rare disease like a specific form of HSP, I just wanted to summarize a little bit about what the field has. And thinking over the last couple decades, at the very beginning, we just had a couple genes. Then, of course, over the last 10 or so years, the number of genes has increased dramatically. So the genetics has advanced tremendously, and it's been a big part of the advance in HSP research. Another big part, of course, is when you have those genetic models, you're able to create genetic insights. You're able to create different models, and those models can span all different types. And you've heard about some of those today. You have these flies or worms. The reason we like those is they're cheap and they're fast, and you can generate them really quickly and make a lot of them for very little cost. Human iPSCs and organoid models you've heard about, those are fantastic because they're human. But as you also heard from some of the questions is, they're not perfect, they're not perfectly representative of the neurons in your brain that are involved in this disease. But there's some similarities, and they're certainly a very valuable system, particularly since they're human, and many of our other model systems aren't. And organoids are where we allow them to grow together, sort of mimicking some of the connections that occur naturally in your brain. And of course, it's not perfect, but it's getting close. And I think it's important to remember with every model system that none of these models are HSP. You have HSP, but a mouse doesn't. It has some features of it that we can use experimentally, but only people have real HSP. Of course, we've heard about mouse and rat models, and I'm going to talk about mouse models. Many of you use rats, and rats are a good model in many ways for HSP because they're bigger. If we think, well, we want to get a long axon, well, why not use a bigger rodent than a mouse? One advantage of using mice, and it's why we've chosen to do that, is it's used for so many other diseases, and companies are so familiar with it. They're very used to mouse models and discussing and doing a lot of the pharmacokinetics and a lot of the toxicity studies. A lot of those have already been done in mice, so sometimes it's easier for us to move some of our ideas into therapeutics a little quicker in that system. But I always used to list just mouse models, but I think it's very important to list rat because I think it is, as you've heard from several other studies and a lot of the work that's funded by the SPF, is that rats are a very good model for HSP, and they have a lot of advantages. And hopefully we'll see more of those in the future. And again, I think that when we have these model systems, then we have these different types of readouts. So if you have a model, you have to see something that's abnormal. In a mouse, that could be that it walks funny. It could be that there's electrophysiologic abnormality. I'm not going to show a lot, but we're starting to use a lot more electrical recordings of the nerves, and that's proven to be very valuable because it's very quantitative, easy to measure. And in cells, we can do the same thing. We can look at an abnormal organelle, or we can look at an axon that isn't growing properly or has all these blebs in it or something like that. So there's a lot of advantages to the model systems that have easy things to see, because then if we treat it and those things go away, we're getting closer to things that might be good therapies for people. So I think that readout is very important. And I think what the HSP field has really going for it in many ways is technology, the technologies of genetics, the technology of new advances in how we do clinical trials, how we measure symptoms in patients, how we even look at cells. And I'll show one or two examples of that because that is what my own work is about. But I think all those innovations, and they're not all specific to HSP, but they're all happening now. And I think the advantage of that is that allows us to rapidly deploy them. And a big advantage of a lot of the technology is that it allows us to do trials cheaper and faster. And that's always going to be good for industry because that's what they're looking for. Now, over the years, and this is not an exhaustive list, I have a couple therapeutic targets that people have tried to go after. And we're doing some of these trials now, and I'll explain a little bit in detail about what we're doing in some of our models, because we really are trying to design our trials in a way that we can partner with industry. And I'll mention a couple things that I think are important in that regard toward the end of my talk. But you can see there's all these different themes that have emerged from all the studies that people have done over the years. And these are things that we can now test in those model systems. We can test them very quickly in iPSC cells. We can, of course, do them in mice. If the mice are already there, we can do these trials. And I'm going to tell you a little bit about how we're doing that, and because there's always a couple pitfalls when you do those trials, especially with drugs that are new. If it's something that's very well known and being repurposed, sometimes there's a lot of knowledge about how to dose those and how often, and and what's dangerous, what's not, all those types of things. But for a new drug, there's a lot of work that has to go into understanding some safety aspects as well as dosing before we can get too far along in the development process and in terms of therapeutics for patients. So again, these are lots of targets, and you imagine we have all these models, all these targets, and people are sort of testing them in different times. So you'll see a lot of the research that you see published in the HSP field is toward this, and it's very important because they won't all work, but the ones that do hopefully can move forward. To human trials eventually. Now, what I wanted to spend a little bit of time on are new therapies, because many of us that were trained years ago in medicine, we're very used to the traditional drug development process. And you heard a little bit about that. You have an assay in a cell, you do these massive screens with as many compounds as you can, and then you sort of whittle it down, you modify them, and eventually you get these drug candidates. Problem is that's very expensive, and for a rare disease, how do you de-risk that for industry? And I'm going to show you some ways that I think we can do that now. But in addition to the traditional drug development process, there's all these new ways we can approach things. We can do gene therapies. Many of you might have learned about that in some of the rarer forms of childhood HSP. They use these viral vectors to deliver into the brain, genes that are missing. So you imagine if you have an HSP, it's a recessive form, you're missing a protein. Well, you might not even need to understand that much about how it works if you just deliver it to the right cells, and it sort of corrects that cell, then that could be a really good benefit for people. The hard part is, of course, it's expensive and it's very cumbersome. It can only be done at certain centers and so on. We do a lot of these for different diseases. And again, you can see some dramatic improvements. So there's no doubt that gene therapy is one of the areas for the future. Another one that's quite a lot easier and can move even quicker is knocking down mutant proteins. So you can imagine if you have a dominant disorder and you have this mutant protein that you're trying to get rid of, if you can selectively get rid of that, then you're going to restore a lot of normal function. And those can be used, things called ASOs, antisense oligonucleotides, there's other ways to knock down genes as well. And these are widely used already in clinical medicine. We do quite a bit of it for other diseases, not just as research, but in clinical care. So that's another area that we can think of for several HSPs. And then a really neat way that we're doing right now in collaboration with some groups at MIT are repairing individual mutations. And imagine if you have a mutation and your amino acid is now changed from one residue to another. Well, what if we just go into the gene and just fix it? That sounds like the perfect solution. Of course, it's hard to get into all the cells, and we don't know if we're fixing it at the right time. Maybe some of the damage is already done. But still, it's a very, very interesting approach, and it's one that we're taking. Now, you often hear the word CRISPR associated with this, but CRISPR isn't as useful for a lot of the human studies because it can have other effects too that are not intended. So there's all these new technologies called prime and base editing, and those are the ones we're using right now, and we're hoping to be able to have some advances in one of the forms of HSP. And I'll hopefully show that next year if we have any results. Now, one of the things that I think we've heard about with the formation of the network of HSP centers is trial design and readiness. And that's really a critical part of this because as we get these ideas coming through, the need to move quickly is very important. Companies don't want to wait, or even academics don't want to wait. And it's great to have all this infrastructure in place. Part of that is also making sure we're not just ready to do trials, but to do innovative trials. And there's a lot of innovation going on now in trial design. And one of the things, I think you heard a little bit about wearables, and one thing we're doing with some scientists at MIT is you don't even have to do wearables anymore. You can actually just put a wireless device in the room, and it can track you throughout the entire house. And it collects hundreds of thousands of data points, and then the machine learning can sort of give you a score based on how you did that day. And it can even follow one person in a crowded room throughout the house. It's almost scary in a way what they can follow because they can see how you're breathing, your sleep cycles, and we found it to be very useful for a disease like Parkinson's where your symptoms go up and down throughout the day. And I'm sure with HSP you'd think the same thing. You could imagine going to the doctor's office and having a bad day, and you're there for your study visit for 15 minutes, and it looked horrible, but a couple hours later you feel great, and how representative was that? So one of the ways that we're hoping to decrease the number of patients for trials and speed of trials up is to collect a lot more data at home. And again, wearables are a big part of that, but again, these wireless technologies are particularly good. We use them in Parkinson's in particular with sleep, because you can imagine it's hard to sleep with a whole bunch of wires hooked to you. So it's a lot easier if you're just sleeping your normal self and there's just a box in the corner that's recording everything. So we do have those. And again, they're a bit frightening when you see how much they can actually do. So the other thing is about innovations in drug development. I think we heard that already. One of the great things about AI is it can do drug screens. But there's also new technologies in a lot of companies coming out where you can just do de novo synthesis. You don't have to screen compounds at all. You can identify a target and build a drug right there. And that has the advantage that it takes out all that screening time, and it makes companies more willing to jump into diseases like HSP that are relatively rare. It essentially de-risks it. You don't have to do massive screens and pay all this money. You can just jump right to a de novo drug design, so you're willing to take on even rarer diseases like HSP. So I think all of those are real good things for our field in terms of the ability to move a lot of the advances that have already been made forward into therapies. I just wanted to sort of end my talk with one example that I'm working on, and I just thought it would be nice to sort of, because I know many of you think about these different mutations, and especially with SPG3A and 4, that can cause these really severe symptoms. And what does that do for us in terms of opportunities for therapy, and how do we use that information even in terms of the science that we're doing? And as many of you know, there's one with this elastin, which is SPG3A. There's certain mutations that give rise to very severe forms. And this is an example of one of those. Again, this is not new, it's from 2008. However, Darius and we worked with him and several other groups a couple of years ago to look at a whole spectrum of these SPG3A mutations. And it turned out that a lot of the kids, mostly kids with these really severe forms, had either the same mutation or one that's right next to it. So I think you can see in this diagram, it's hard to read the individual things, but there's a couple important themes from that paper. One is that almost all the mutations are missense, one amino acid swapped for another. So that tells us there's a bad gene that we need to get rid of because it's only on one, it's a dominant disorder. So if we can get rid of the bad one and we think that one is enough, then we've made a significant inroad into therapy. So when we think about a disease like this, when we see it in the context of all the other mutations, it gives us several therapeutic possibilities. First is it might just be a more severe form of the other one. Maybe the same drugs will work for both. So the typical drug discovery. maybe we can use an ASO and just remove the bad allele, or remove both of them and just get it down to a point where symptoms are better, and that's possible. Or there's the gene editing approach where we go in and fix it. So that's what we're doing. We've created a model that I'll describe that is sort of humanized. It's a mouse that has some human features, not a chimera, not like a mouse that looks like a human, but a mouse with some DNA that's human DNA. It's not as sort of Frankenstein-like as it sounds like it might be. So what we do is we pick the HSP proteins, obviously that are related to ER function in terms of our studies. And the reason we need to combine them is when we try to model a disease like HSP in a small mouse, we're missing one big feature, the length. How can we model a disease of 1 meter axons in a mouse that's only 10 centimeters, so much smaller. And one way is to combine different genes that work in the same pathway and get a more severe phenotype. And we've done this over the years, and you can see, here's an example. This is not a new paper, but I can show you the mouse at the bottom has two HSP genes mutated, and the one at the top is completely normal. The good thing about this is the mouse develops its abnormal phenotype very young, and therefore it's something where we can start screening right away rather than having to wait a year or two. So again, having a good mouse model, this is just some of the quantitative aspects. The only thing, you don't have to read this, the top is this males, the bottom are females. It's just a couple different assays. What that allows us to do is compare mutant to wild type animals. So wild type are the normal ones. And what this allows us to do also is we can start treating these animals, and we want to see the bad findings become closer to the normal. And one of the things we've decided to do, so this is a mouse model that we've already developed, but we're doing the exact same thing with that rare severe mutation in elastin. So this is a regular mutation. The new mouse is the same basic idea with a severe mutation. And it looks like it is more severe so far. Another thing we see is that when we actually look in the spinal cord, in the corticospinal axons, which are of course the ones involved in this disease, we see a huge abnormality in ER, which is kind of good because we said, well, it's an ER protein and ER morphology is disrupted. And in our mouse model, we see a huge effect. So the one on the left is a normal axon going kind of up and down, sort of, it looks like northeast to southwest. And then the other one is just going north to south on the right. And you can see that there's these little lines going horizontally that aren't present on the left. And it turns out that that's abnormal ER. And to do this, we had to use a technique called FibSim. And as I mentioned, one of the advantages we have in this field is technology. We have these new ways of looking at things. So we can actually use a very special form of electron microscopy. It's only available a few places, and we're able to get a very high resolution image of the ER. You can see in panel B there, the white is what the ER looks like when we do it this way, and the green is what it looks like under a microscope. So you can see the microscope misses all sorts of detail. So one of the important things is as we move forward is to make sure that we have the sensitivity to see the abnormalities that we need to. And everyone likes to hear about AI, and this is one way we use AI. We used this like 7 or 8 years ago. None of this is new. But this was back when we told AI what to do. Now, of course, anyone that works with electronic medical records knows that the computer tells us what to do now. But back then, we actually used to be able to train it, and it followed commands. So we actually asked it to go through all these massive data sets, find us the ER, and reconstruct it. And it was interesting because the abnormality was so great, the computer couldn't do it. So we had to keep going back and training it. And eventually, it got it and was able to reconstruct. So this is what they call segmentation. So the computer has to identify the ER. And it's a lot harder than it sounds because microtubules are the same diameter. So it's hard to tell those two apart. So people would go nuts trying to do this. But what we're able to do is we're able to create massive numbers of these data sets, and the computer can go do it for us while we sleep at night, and the next morning it's all done. So, and this is what it gives you. This is a completely 100% computer-generated figure. No human being did anything to this. Of course, now we get in trouble with ChatGPT if we do this, but 6 or so years ago when we first did it, it was considered an advancement. Now it's considered cheating if you do it now. Again, you can see the difference here. On the left is a normal axon, and on the right is the abnormal one with the mutations, and you can see there's all these like little ladder-like structures. So very easy to tell. And it's a great readout for us because what we're able to do now is we're able to not only look at the phenotype our mouse has, which are the abnormalities that we see when we examine them, but we also can look at their spinal cord. So when we treat them, do we make those ladders go away or not? And that'll be an important part of our therapy. Another new technology, and I just have 1 or 2 more minutes to go, is the ability to see individual proteins. And one of the challenges in biology has always been, we're trying to look at something with the tools that we have, and often they're not good enough. But we've been able to work with several groups. We're very fortunate to have several collaborators who are really the world leaders in this, and they're able to allow us to see an individual protein. And they're not only can we see it, we can see it move. In real life. So this is an individual protein moving. So you can see there's all these different proteins moving on the ER. And they're moving so fast, we usually have to slow these down. And why this is relevant is here's an example with spastin. On the right is a mutant form, and on the left is a wild type form. And that's one spastin molecule moving. And you'll notice on the left it moves just fine. It's going along the ER. The one on the right is moving so slow you might not even know it's moving. And that's what the mutation does. And when we think about how all these different mutations might give different symptoms, one of the things we're looking at is not just thinking statically, but thinking dynamically. Maybe part of the problem is that the protein isn't moving properly. And that's going to be a new line of investigation. So you can see, look at the huge difference that is. So you can imagine when we start doing therapies, we can see if it's changed or not. And here's another one that we just published. This is an ALS. It's a related disorder, of course, not HSP, but some similarities. And we see the same thing. At the top is a normal ALS protein. It's called VAP-B. And at the bottom is a mutant form. And you'll notice the mutant form doesn't move as much. So we have the ability now to use technology not only to improve the trial designs, the things we do with patients, but also to improve our sort of trial design and analysis of cells. So this is a whole new, I think, technological advancement. And we've started using this, what we call it, sort of dynamic systems, a lot more in our HSP analysis. And we're doing it in HSP and ALS, and of course the natural disorder to add in between is PLS. So we're starting to look at PLS models because we know that those have historically been much harder to make because there isn't as much genetic understanding. So we're starting with patient cells and really trying to see, are there abnormalities we can see in PLS, to really sort of jumpstart that field in terms of some of the models. But again, these are huge, huge effects. So we're really hopeful for the future that this will be part of the design of projects going forward. So again, when we think about kids like this, one of the things we're doing is we're creating this new model system, but one of the reasons we decided not to do it ourselves and to work with a lab called Jackson Labs, which is buildings and buildings full of mice. So that will give you nightmares tonight if you think about it too much, but there's like a whole city of buildings that's just filled with mice, and they're experts at it. And what they're doing is we decided to have them make our mouse and with a human portion around the mutation so that we can do gene editing, but we're also doing drug trials and we're having them do it in the same way they do it for industry. So we're going to learn a lot about what they think are good ways to analyze HSP models, and of course we'll share that with everybody because that might be a really good way for us to know what are the best types of measures in these model systems that allows us to interact more with companies that might be interested in developing some of these therapies. So with that, just wanted to emphasize that, we obviously, science is a team effort. My own group, because I do a lot of administrative work, is very small, but we benefit tremendously from collaborations throughout the world. Certainly I'd highlight Darius. We've worked with him for many years. He's our neighbor across town. David Liu's lab at MIT, he's the sort of the pioneer of prime and base editing, and Jackson Labs, of course, does all of our mouse work in collaboration with us. And finally, at the bottom there in bold, Jennifer Lippincott-Schwartz is really sort of one of the pioneers in cell biology in the entire world, and we've been very fortunate to work with her the past 15 years or so, working on models of disease and analyzing them. And again, it's been a tremendous benefit for us and hopefully for the field to have that opportunity. I think I have a few minutes left if anyone has any questions I can answer, and thank you.
Okay, we've got time for one. We're going to do one right here.
Thank you. So You mentioned physical therapy. I've had a couple of physical therapists tell me that given my disability and the aspects of it, that there is no benefit to doing it. I've been told, well, stretch your legs, it'll feel good, but your muscles will contract back to what they were.
That certainly hasn't been my experience in the patients that I've seen. I think they've benefited from it tremendously. And you could ask the people around, they would know around here in the room, they would know better. I think it really, because I think that, your body is plastic in the same way that if you're an athlete and you're stretching, that benefits your sport. I don't think it's any different for somebody with HSP. It's going to benefit you, and it's going to prevent you from being injured. It maybe, hopefully, will decrease things like pain. It might not slow the underlying disease, but the symptoms that you feel will be better. I think, again, there's lots of doctors here you could ask this to, but in my experience, it's better than the medicines that we give. It has fewer side effects, and I think it has a real measurable impact on people's lives. At least that's what I've seen in all the patients I've seen.
Thank you. Yeah,
Sure.
I just think they were thinking the spasticity and the fighting back, or that they're, I don't know, but
Well, remember, they're not used to,
multiple physical therapists
they're
told me
not
that.
used to HSP. That's the thing, is a lot of people don't have experience with it. And
You're very right.
they'll think it's the same as a stroke or somebody with cerebral palsy. They'll sometimes lump you into those groups, and HSP is different. So I think it matters.
Can I ask one other question?
I'm going to have to give somebody else one more chance because we got to move on. Any other questions real quick? We got one more over here and then we've got to move on. It's a big day. Here we go.
Dr. Blackstone, it's good to see you again.
See you.
I was in the undiagnosed disease program in 2012.
Yeah, it's a while ago, yeah.
And I was tested extensively at the NIH Clinical Center. And you were on the team.
That's where I was before.
Do you remember Colonel Saburo?
Yeah, we used to see quite a few people with HSP around then because, again, that was at a point where it was really underdiagnosed and people weren't getting the testing. So people like you would come through the undiagnosed diseases program at NIH. And now, hopefully, those diagnoses, people can get that in their doctor's office. They don't have to come all the way to Bethesda.
Your clinical diagnosis was spastic paraplegia. Felt to be consistent with, consistent with hereditary spastic paraplegia of unknown type. So the question is, that was 12 years ago. How many new types have been identified since then?
Well, there's a lot of new types since then. I don't remember exactly how many are in that year, but certainly, around the late 2000s into the past 2010, a lot of the new next generation sequencing techniques were coming, and insurance started paying more for some of these tests. So I think over those 5 or 6 years, there was a lot of new genes identified. Plus, people were looking more throughout the world, at other populations and so on. But it raises the point that there's still a lot of people out there, and I think the percentages differ from study to study, let's say 30%, who we really believe have HSP that we don't have a genetic diagnosis for. So I think it does emphasize the importance of those continued genetic studies and really bringing centers together to find new genes, because I think that's one of the key steps in terms of therapeutics, is knowing what type you have, because that might alter the direction that you go in terms of treatment. So I think it's very important that obviously this network, one of the real focuses it's going to have is trying to shrink the number of people who don't know what form they have, and so we can find more forms and give more people answers to what type they have.
Great. We got to go. We got to move on. Thank you very much. Great questions, everybody. Dr. Blackstone. Okay, guys, we're trying to keep you on time and get you going. We got a lot more to do this afternoon. All right. We're going to get up here. We're switching everything. We're going to get you set here, Dr. Mitsumoto. There we go. There we go. Short. Good. Yeah, I know. All righty, here we go. We're ready. This is the quickest change we've had all day. Dr. Mitsumoto, everybody.
Thank you very much. Thank you very much, Dr. Gladstone. Switching with me, I had unavoidable conference call. So I'm so happy to be here, and this foundation is an incredibly great organization, and also I'm grateful for your continued support for our study. I'd like to talk to PLS today, natural history study. Let's see. Oops. Okay. Just a minute. So before talking to PLS Natural History Studies, last year in St. Louis, I was at this meeting and sitting together one table with a patient with PLS, and they asked me about the startle reflex. Startle reflex is something, noise or someone talk to you, suddenly jump up. And that's called the startle reflex. And one of them asked me, do you know anything about the startle reflex in a patient with the PLS? I said, no, I never heard. And then he started asking around. Turns out most people who were there had a startle reflex. Some are a lot of startle reflex, some are some. So wow, I did not know. That was the beginning. I decided to ask our patient natural history studies, about 70 patients, and most of them had surprisingly startle reflex, which was not described in the textbook, and I have to admit I did not know. So I asked our coordinator to start this startle reflex, and then looking at the ALS, which I am specializing in, and patient with ALS doesn't seem to have this startle reflex. However, a couple of patients had startle reflex, which are predominantly upper motor neuron dominant ALS. So actually talking to us and suggesting a doctor to initiate some surprisingly new studies, and eventually we're going to write this up. Now, today we are talking about a little bit of history among ourselves and natural history studies, and another conference international conference will be held in this December. So in terms of our perspective histories, PLS already we heard many times, which has been rare disease, puzzling and obscure disease, just so rare, and considered to be an extreme end of the ALS spectrum, and neglected disease. We conducted this prospective 5-center PLS Cosmos studies sometime in 2009 in conjunction with NIH-funded ALS Cosmos studies. Cosmos means actually oxidative stress multi-center studies. We decided to develop PLS-specific functional rating scale, which we're going to talk about later. And also, we had the International Conference 2019 in Philadelphia, and conference supplement was published. PLS diagnostic criteria was also published after this conference. And PLS natural history studies initiated based on this conference. And now, next third PLS conference, international conference, will be held in December, as I told you. So the first disease-modifying clinical trials in PLS is not any longer unrealistic and far-fetched. But I heard Dr. Gladstone talk today just before me. I see for HSP clearly light in the tunnel. So I think we should be very hopeful of PLS. Now I'm talking about HSP, we are talking about the PLS. Phenotypic and molecular analysis of primary lateral sclerosis paper was published based on PLS Cosmos study. So this study taught us something very unique, very important. ALS FRS is everyone who does anything with ALS using this ALS FRS, which is the standard, most commonly used functional rating scale. When you look at ALS studies, usually runs about 1.0. Take a look at these red letters and run 0.9, 1.2 monthly, moving pretty fast. ALS moving fast. The functional rating scale was made based on ALS. So we used this ALS-FRS for PLS studies. It moved monthly 0.05, which is more than 20 times less, actually, moving so slow. So we cannot use usual ALS-FRS for PLS. That's the reason we decided we need some PLS-specific functional rating scale. So comparing ALS-FRS revised, everyone using this for ALS, some people using for PLS, but we use the PLS-specific, more interim grades included and used. So based on this scale, we can show disease moving pretty well in PLS. So we have to now demonstrate it is also true in natural history condition. So also we made a consensus diagnostic criteria for PLS. There was no really consensus diagnostic criteria in the past. We published this based on this conference call. And natural history studies also planned. And in natural history in the past has been mostly retrospective at the best. And our previous application of an NIH application, we presented because we need a NIH grant which funds a lot of money, and yet we was not approved because we don't have any natural history for PLS. That's the beginning that we need to think we need such studies. Rare disease such as PLS requires a multi-site study because so rare, just we cannot do by one centers. We need more centers. United States, Canada may not be enough. We have to move to Europe, Asia, and Australia. Natural history is a prerequisite for designing future clinical trials in PLS. We need to determine the best outcome measures for PLS clinical trials. We should be able to validate the reliability of new diagnostic criteria. And also to improve understanding of PLS is very important. To develop biomarker for the futures, a lot of purpose, a lot of objectives. So we got the multiple funding, of course, SPF and ALS Foundation, Mitsubishi Tanabe, so forth, and private donation from Mr. David Marin and the family. We got those funds to start, and we organized the 31 centers, all those specialized PLS specialized people, investigators. And then those happy face site for the United States and Canada. A couple of centers cannot continue, but about 29 centers continue to do this natural history studies. This is the organization. Columbia University is organizing centers. We have committees, executive committees, and EMG. I'm going to talk about all those committees organized studies. And standing committee and objectives, steering committees made of those people, and other committees I'm going to talk about. Any issues needs to be resolved by steering committee. And inclusion criteria of the studies, people has to be older, 25 and older, and the diagnosis has to be established by new PLS diagnostic criteria. Symptom onset no more than 15 years prior to the baseline. And also ability to independently walk, because we have to measure the speed of walking. Doesn't matter whatever assistive device you use, not wheelchair, but we has to be able to walk. And in case where molecular testing has been done, there are no HSP or HSP-related mutations, and expected to have at least some bulbar symptoms, because most PLS patients develop some bulbar, means speech and swallowing difficulties. And then if we include those people, the possibility of clinically HSP is becoming less and less. and upper motor neuron symptom signs in region other than legs, and try to exclude the patient with HSP. Normal brain scan, spinal neuroimaging has to be normal or consistent with the PLS. No active major neurological and other medical disease, and residing in reasonably commutable distance, and no history of ALS or PLS in immediate family, and no family history of hereditary spastic paraplegia, and examination less than 12 months has to have EMG, but we modified because this excluded a lot of possible patients, so we allowed a little bit more easy way. So those are inclusion criteria. There are some exclusion criteria. Unwilling to unable to give informed consent, upper motor neuron symptoms and signs only in legs, and unwilling to unable to visit the study site clearly exclusion criteria. And clinically obvious cognitive impairment needs to be excluded, and participate in clinical treatment trials not a candidate for this study. So recruitment enrollment, and we decided to recruit 50 early PLS. Early PLS including less than 24 months after symptom, also including probable ALS, based on current criteria, consensus diagnostic criteria. 24 months to 4 years are probable PLS. Then what to do with 24 months? Upper motor neuron, less than 24 months after symptom onset, we decided to include. I strongly believe myself, disease has to be early. The time take more and more, neuron degenerate, axon degenerate. The best time is to treat is earliest time. So we need to include a patient with early stage of disease. But also 50 patients are definite ALS. That means 4 years more than, 4 years after symptom onset, but less than 15 years. So those are inclusion criteria are early and definite PLS. So a lot of studies, including baseline, 3 months, 6 months, 9 months, 12 months, and 24 months. A lot of needs to be done, including cognitive and also testing. But this study is very unique because we are using via phone, iPhone, whatever phone, using those phones to get measurement and histories, status. But the baseline 6 months and 12 months has to visit the site because they have to check upper motor neuron signs by physician, and also timed up and go has to be done at the site. So this seems to be all the slides set. Sorry, I made a mistake. But anyway, we published paper, this natural history studies for protocol. So if anyone wants to see more detail, you can look at this paper. So characteristic of PLS natural history studies, this is a large multi-site study recruiting a participant with extreme rare disease. It is a study funded by multiple funding agencies, a donation, and Neurobank. Neurobank is based on Mass General, and they donated their time, equipment, everything for these studies. And the study is managed by steering committee and other committees. It is a study which most common data obtained by Columbia Study Coordinator Center by smartphone. The study is a prerequisite for the future clinical trials in PLS, and also it is incorporated with future genetic and biomarker studies, hopefully expanding our understanding of PLS and pathogenesis. So we explore biomarkers. We are looking at oxidative stress biomarker in the urine, lipidomic studies, and microRNA changes. Those are planned and being, and just we got early oxidative stress seems to be no difference compared to controls, which is very important negative studies. We also analyzed DNA exome sequence. Dr. Matt Harms and NIH Brian Traynor are involved with a genome sequence, not exome, genome sequence, looking at some underlying genetic cause of PLS. So biostatistic analysis will be done by Dr. Ken Chan, Columbia University, and looking at analysis characteristic natural history of all 2 patient cohort, and providing information useful for next clinical trials. Primary outcome is PLS FRS, whether this is good enough to use future clinical trials, and longitudinal outcomes such as PATACA and NeuroQOL, finger tapping, slow vital capacities, and TUG, which is which I mentioned, walking distance 3 meters from sitting position, walk 3 meters, coming back to original chair and sit down, excuse me. And also we have site visit 3 and 6 visit with phone call from baseline. And electromyography is done at 12 months. So outcomes, I mentioned all those outcomes, including cognitive impairment, and Penn upper motor neuron score and timed up and go. TUGS test has to be done at the site. And EMG committee checking all EMG baseline done at doctor's office or at the site, and they approve patient can be enrolled in these studies. And also at the 12 months, all do exactly same EMG, same muscles, evaluate changes based on Gold Coast criteria, which is ALS. ALS criteria is very helpful to determine who has ALS, but PLS is very helpful based on this criteria who does not have this ALS sign. So EMG committees, as I said, looking at all those changes baseline and at the 12 months point. So this data will be so important how we define at the baseline as a PLS, and also 12 months how disease evolves, whether it remains to be a PLS or any sign of ALS. Parenthetically, I should mention, if PLS is becoming a little bit more ALS, upper motor neuron initiated disease, even becoming a little bit ALS, clinical features would not change, remains to be slow, remains to be clinically PLS. So diagnostic validation committee is these people all among our sites, and also publication committee looking at any papers they read, they comment to make sure our paper is within our guidelines, purpose, and objectives. So we had a serious concern about execution of PLS natural history studies. This is large multi-site studies. It takes an unbelievably long period of time to obtain IRB approval and set up a subcontract with each site. So multiple site, 31 site organization is very, very difficult. And furthermore, we had COVID. I'm sure everyone wants to forget about the COVID, but the COVID stopped our study tremendously. And the patient cannot come to the site for enrollment, and the people are not coming to the institutions. Even administrator, people, ourselves cannot come to so easily. So delayed just enormously. Yet we have a coordinator. We have to spend some certain money we have to spend. The study initiated 2020. The money is spent. Of course, study cost would not be spent because the patient is not there. But because we had a shortage of funds, and thanks to SPF, and then they supplemented some deficit, which was greatly helpful to carry on these studies. So we have had enough time budget to conduct the complete studies as the organization supported. And we had aimed 100 patients, but became so difficult that we stopped at the 72 patient because we at least reached 2/3 of objective. So currently we have now overall 72 patients, and average age 62 years. Male 40 and woman 32, and disease duration 5.5 average. And then early means less than 2 years, 5 patients, as you see, and probable, that means less than 2 years, but longer than 2 years was about 25. And the definite is longer than 4 years, 42 patients. Currently, those are total of 72 patients. We will finish entire studies end of October, and hopefully we can analyze some data based on Ken Chun, and we can report at the next meeting in December. So next major question is, we have already planned to obtain outcome data, PLS, which is the primary outcome, and other obtainable data via phone during a 24 months visit. Some patients have already completed the 24 months telephone visit, and probably more than 65 patients still has not reached the 24 months. And the natural history study is the first and only study looking at the long-term natural history of PLS, at least 24 months. But this is incredibly important study, and our database and people suggested we should keep going to obtain long-term, at least 5 years, long-term health condition, the PLS conditions, how they do long term. So based on those expressions, we aim to extend 5 years of follow-up studies, and probably I'm going to ask our colleague, Dr. Lee at Columbia, the successor of these studies at this point. So I'm going to switch a little bit. We are going to have third international PLS conference at International Continental Hotel, Intercontinental Hotel, Montreal, December 4th, 1 to 9 p.m. So we will have 31 speakers, organizers, and we have 12 members of program committee helping to establish this speakers and panelists and so forth. This is very unique about the several panels, only several panels, although Dr. Beatty, Peter Beatty is a keynote speaker and talking about the neuroimaging in PLS. And probably anyone who is involved in PLS, you know, neuroimaging most rapidly progressing. So we are going to hear what Dr. Peter Beatty say at this meeting. But rest of the meeting are made of all panel discussion, unique because people can present only three slides, and 5 minutes talk, and maximum. And so everyone talks, and I also believe a really short presentation to the nutshell, really key point is important. to spend more time to discussion. So we are talking about diagnostic validity of current criteria. We made the criteria based on consensus. How accurate, how reliable is very important. That's a panel discussion. ALSFRS and other outcome measures are key for future clinical trials. We are going to discuss in panel discussion. PLS registries, repositories. Now, PLS is, I learned, United States, Canada is a vast, big countries. You think enough patient to do clinical trial, natural history studies, turns out to be very difficult. And some people are living very distant from centers. People cannot come, even using a telephone is very difficult. We need a larger population. And we know about roughly 2,200 patients annually present in this United States, but not good enough, not large enough. We have to expand study population, patient population to Europe, Asia, and Australia, and perhaps, I don't know, South America or not, but I think we need to expand so patients are available for clinical trials for the future. So that's we are going to discuss ALS registry and biorepositories. We are talking about biomarkers. We are developing biomarkers, not perfected yet, but clearly some future possibilities. And rest of the times we are talking about the clinical trial, how we design, how to develop, and also steps towards the next clinical trials. That's the major goal of this conference. So anyone who is interested in, please come to this meeting. We also make copy of this conference and record, so anyone can look at, participate later. You can look at this recorded conference. So again, already the conference, international conference supported by SPF, MNDA, British MNDA Association, Mitsubishi Tanabe, Niels, and also Mr. Marin and the family supporting this conference. So I am quite excited about conference to look over, make PLS more being aware, and also making progress in the future development. So thank you very much for your support, and I really appreciate it. And if you have any question, I'm happy to answer if you approve.
Yes, sir. We got a few minutes. Great job. Congratulations. Dr. Mitsumoto, Hank Chupi, who is watching on the live stream, asks, what is the difference between PLS and upper motor neuron, ALS?
Well, ALS is a combination of upper and lower motor neurons. Upper motor neurons reside in the brain, send tracts to spinal cord to give a signal to lower motor neuron in the spinal cord. Based on that signal comes along the arms and legs, bodies, to the muscles contract. See, I want to move. So that's the disease, upper and lower motor neuron as ALS. PLS, upper motor neuron only. Somehow, lower motor neuron and spinal cord is spared. Why? They are resistant to the disease, or they have nothing to do with the PLS disease. Whatever the reason, spinal cord motor neurons are totally spared. Disease, upper motor neuron. So your question is, what is upper motor neuron ALS? Disease we call ALS is not just one single disease, uniform disease. Everyone agrees ALS is multiple disease, up and orange and other things. The disease we call ALS. Some disease are predominantly lower motor neuron, a little bit, well, almost no upper motor neuron. Some diseases are predominantly upper motor neuron, very little lower motor neuron. That's what we call upper motor neuron dominant or lower motor neuron ALS. Upper dominant ALS, it happens to be better, clearly better, slow, going on years. It's very similar to PLS. So what's the difference PLS versus upper motor neuron? Of course, we don't know. But if we know PLS, why lower motor neuron are spared, why upper motor neuron only affected, if we know that secret, we can do something, good things to slow down or help the disease. So we need to more understand the disease. Basic science is so important. But I am a clinician, so looking at the patient, how we can help. And of course, at the moment, we don't have any medication to treat. And Dr. Handy is showed this morning as a keynote speaker, and a possible medication presented. It is likely. But I think we need to understand how patient and moving the disease. So if medication is available, we can immediately try those medications based on natural history studies. Thank you for your question.
Great answer. Great question. Great everything. Sorry, I end up over here. Sorry about that. Here you go.
You might remember, Dr. Matsumoto, I was attending that medical conference when your whole study started, and there were genius PhDs in the room, all experts on PLS, and then during dinner or lunch, I don't remember, someone came up with the question is PLS a form of ALS or is it a unique disease? And everybody voted and spoke independently, the different scientists, and about half of them said it was a form of ALS, and about the other half said it's a unique disease. So after your whole study, have you come up with a better conclusion?
Of course, answer is not yet.
Okay.
But I think ALS, PLS is PLS is extreme end of the spectrum, ALS spectrum. So interestingly, if you look at autopsy, spinal cord, and so forth, and looking at more biomarkers which are available currently, and PLS has a features of ALS. Looking at the cell, there is a changes found in ALS there, yet not clinically obvious, clinically spared, but morphologically, pathologically, there is there. So I think disease is spectrum. And as I said, ALS is not single disease. PLS is probably not the single one disease, multiple disease in it. We are not good enough to separate out. Seems to be just we call ALS or PLS. So, looking at it, I know everything, but it's not true. Still so much more we know, we realized how little we know. So we have to do a lot of studies need. But as I said, for this foundation, I see the light in HSP, clearly light. So you have to go after it, go after that area. PLS is a little bit way behind, but still we are moving. So I believe we are right directions.
Thank you.
We've got time for one more. I'm going to get one over here because we've got to get you guys moving to your next stuff.
My doctor told me that the difference between ALS and PLS, we can have all the same symptoms, but the difference is ALS, your muscles are completely destroyed. With PLS, it's weakened to nothing.
That's correct. Big difference between both upper and lower motor neuron and a pure pure upper motor neuron. And, you know, lower motor neurons are involved. People develops respiratory problem and significant bulbar signs. And so combination of upper and lower motor makes the disease much, much, much worse. PLS is a pure upper motor neuron. Yes, without any doubt, people have tremendous impairment, difficulty walking, difficulty using arms and so forth, but much slower pace. So I published a paper a couple of years ago hastening the diagnosis of ALS. ALS is a dying disease. Most people after diagnosis dies within a few years. Well, some are longer, but that's true. But still chronic disease. Chronic disease, physicians are relaxed. Relaxed, making a diagnosis quicker. We don't have any better medications. So you see the doctor. If you call to see doctor, they don't see you next day, next week. It takes 2 months, 3 months sometimes, average. So you think disease would not progress too much in 2 or 3 months? That's not very true. That's not true at all. For chronic disease like ALS, disease is progressing. Time is essence. The data is shown, the clinical trials and medication, which works a little, but works medication, if you delay, benefit is much, much less. So we have to start the medication sooner. Now, PLS, even of course, HSP, diagnosis not made immediately, slow. And disease would not change. I think that's wrong. I bet the future, when I die sometime soon, and future, people realize chronic disease, ALS, PLS, HSP, Parkinson's disease, Alzheimer's disease, not like stroke. 90 minutes a key. Doesn't work that way, but still you have to make sooner diagnosis and treat faster. Best example of spinal muscular atrophy. So spinal muscular atrophy and SPA, and we used to call Wernicke-Hoffman disease. That disease, the baby die within 12 months, or at most a few years, baby. And now genetic disease treatment is available. If you start earlier, you can stop the progression. You can delay the progression. So the time is essence. And so make sure we have to tell the doctors who are young doctors proud. My first patient is waiting list 3 months, so much request coming. That's wrong. You have to see patient as fast as you can because you cannot deal with slow neurodegenerative disease and slowly. You have to move faster. So that's the reason we published the paper. I think can be applied to PLS and HSP. Sooner is better. Sorry. Okay.
Okay, everybody, thank you very much. Dr. Matsumoto. Okay, we got some stretching coming up real fast, but you got some complicated moves you need to make here after Zach gets done with you guys in a couple of minutes. Remember, we got the small breakout sessions coming up. The first one with the caregivers is actually going to be right down this hallway, just out and around and down in this left-hand corner over here for the caregivers. For the women and the men, you've got to go all the way up to the front corner of the building on this floor, and there's an elevator to take you up to the second floor to some breakout rooms. It's going to be a little tight. It's going to be a little time consuming, okay? And there's a staircase for people who can use stairs, and there's an elevator that only goes up one floor, okay? And so we'll own it. When you get there, it'll be your elevator, okay? So that's you guys are, it's completely on the opposite end of the building, on the front corner, opposite of registration where you get your hotel keys and stuff. Upper right-hand corner over there. You got it? Okay, you know where you're going, that's what's going on, and then after, the only group staying in here is going to be the parents of childhood onset with Dr. Fakari, okay? That's the group that's going to be staying in the room for their presentation, okay? So Zach, you got to keep them moving, you got to get them going. You're the man, here he comes.
Thank you for those of you who are going to stay with us. I hope not too many people are going to get up.
Come on up. You can just grab that in your pocket if you want to.
Yeah. Thanks. Okay, so there's something I just wanted to note before we start. This thought has been just moving through my head all day. And something I've kind of realized through, sorry, I'm a teacher and people moving is very distracting. No, it's okay, it's okay. So something I've come to realize with having HSP and kind of what I hear other people say is just because a cure doesn't exist does not make positive progress impossible. Positive progress is always possible. I mean, as you guys can see from the back of my shirt, it's something I thought of. The only disability is believing you have one. We all have the ability to positively progress, no matter what we hear anybody say. It is always possible. So before we just started, I wanted to let everybody know. So, oh, please applaud yourself. So let's just start with stretching out our hands. If you're sitting down, great. Let's just bring our arm right out and try to pull our palms down. And if you can't pull your palm, you can just grab a thumb or you can grab one finger, whatever works best for you. Because I've come to realize through boxing and Muay Thai, which I've started to do recently, let's switch sides. I feel like the story when I was growing up was that I have a degenerative disease and I can't really progress positively. Let's switch sides. But then I realized I can learn and I can grow. And boxing and Muay Thai kind of helped me see that. And let's switch sides one more time. Because every day I left, I was able to leave with new knowledge. I can guarantee you my hips have never felt better than when the days I do Muay Thai. Okay, let's bring our arms straight out in front. And we're just going to spin our hands. I saw this video on Instagram where it's impossible for somebody to move one hand forward and one hand backwards. If anybody can do that, very cool. All right, let's go backwards or switch the rotations, whichever one you guys have been doing. Doesn't matter which one you do. And let's switch sides. All right, everybody. So let's shake out our arms. Do what you gotta do here. Okay, so we're just gonna start with pumping our arms straight up in the air, not doing anything crazy. You can turn it into a dance if you'd like. Okay, now I see a couple new faces in the crowd, but before last time we did a little bit of boxing, I'm gonna do a little bit again because I did see a lot of smiling faces. So for those of you, everybody can sit like we did last time, but if you want to stand, totally feel free. So for those of you who are not here, I'm just gonna start us again real quickly. I'm gonna say numbers 1 through 6. When I say 1, you're gonna throw your hand opposite from your dominant hand. So my dominant hand is my right. When I say 1, I'm gonna throw a left hand straight out. And when I say 2, I'm gonna throw my right hand out. So once again, let's just throw 1 and 2. Nice work, everybody, nice work. You might feel your shoulders popping. Let it, it's all positive. Okay, now for those of you who remember, we can go right in, but we're gonna do 1, 2, and 3. So our 3 was when we took our left hand, or the hand that's not your dominant hand, and we're going to bring it we're going to hook it to the side and just end right in front of us. And then 4, the next one, same thing with the other hand. So let's just do 1, 2, 3, and 4. So we're gonna do those all together. Let's do it. 1, 2, 3, 4. Just throw a couple, and then we're gonna add in 5 and 6 in a little bit. Nice work, everybody, nice work. Okay, so actually I'm gonna challenge you guys a little bit. We're gonna throw a little combination here. Let's do 1, 2, 4. Let's see if you guys can do this. So if you're doing it along with me, where you're throwing your left hand as 1, we're gonna go 1, 2, then we're bringing our right hand back, we'll go with the 4. So we're going 1, 2, and with that same hand you just brought out, we're bringing it right back to the side. These ones were the most difficult for me to learn, using the same hand. Nice work, everybody. Now let's throw a couple. 1, 2, 3. This one is going to be a little easier because it's just alternating the hands we did. So we're going 1, 2, and then 3 with the hook. 1, 2, 3. I want you guys to throw about 5 to 10 of these. Then we're going to reconvene. Nice work, everybody. Nice work. See, positive progress is possible. I don't know if not everybody who's doing this with us right now might not have HSP or PLS, but as you guys could see, over time, we can grow. It might hurt. It might be very painful because I can almost guarantee you it does hurt. It is possible. We're not done. Just wanted to get that out there. Okay, so now I'm gonna add in our 5 and our 6. So our 5 and 6 are uppercuts. So this is where we're just going to bring our hand like a shovel. From the bottom, bring our hand from the bottom and straight up almost to your opponent's chin. Okay, so we're gonna throw 1, 2, and then 5. So what it will look like is this. So we're throwing a jab, across, and then an uppercut with that left. And we're gonna throw a couple here. Nice work, everybody, nice work. Nice work, nice work. Okay, and I just want to get a little bit of leg movement in there. So for those of us who can, and if not, do what we're going to do with our legs with your hands. So what we did last time, let's just shake them out. Do what you can. If you're having trouble moving your legs, no worries. Just give those arms a whole big shake. Okay, and as we do a Muay Thai, thank you.
That was really great, everybody. Thank you. Remember, you've got other places to be right now, okay? So you got to get yourself to the other breakout sessions that you're supposed to be at. Women only, men only, none of that mixing stuff, okay? We got guards at the door to make sure that doesn't happen. And then the caregivers are just right around the corner, down in this corner over here to the left. So get to where you got to go, and we'll see you for another presentation or dinner later.
Well, good afternoon again, and you continue to hear a lot of great information. Dr. Mitsumoto, Dr. Blackstone sharing some of the more recent concerns and more recent research that everyone is working on, and that's all good to hear. With me now, though, another one of our sponsors, another one of our partners this weekend. I have with me Leah Fronerith with Canine Companions. And you guys know these animals have been so important, these beautiful dogs, companions and help to many of our members in the past. We have at least 2, I think, who are here this weekend with this group. And so what Leah and Canine Companions do, not only here but all over the country, maybe all over the world, you can share, is so important for our community. And so Leah, thank you for being with us. Thank you for your Canine Companions, what you guys do. Let me just ask you to share what you guys do here locally, what maybe you do across the country, and how you see it kind of a ministry to and a help to this group.
Absolutely. So Canine Companions is a national nonprofit that trains service dogs, hearing dogs, and facility dogs for individuals and institutions to utilize and help individuals empower themselves and gain independence. Our dogs are found at 7 different regional sites across the country. Applicants can put in their application and tell Canine Companions what they're looking for in terms of a dog, what services, what tasks, what independence someone is seeking. And Canine Companions will work on finding the right match, finding the right dog, and place that dog free of charge for individuals with any kind of disability that we can serve.
Can you share some of the specific functions that one of these animals could do for someone that might have HSP or PLS that has mobility problems, getting in and out of beds, through doors? Share some of those kind of things that the animals can assist with.
Absolutely. So our Canine Companion dogs have several tasks, 45 plus, that they are trained with upon graduation. It can be taking and tugging an arm of a sleeve of a jacket off if that's a difficulty for someone. The dogs are trained to pull those things off, pull off of a sock. They're trained to retrieve items that are on the ground and hand them to their person or to the registrant at the cash register. They are trained to alert people in the household if there's a need from an individual that is matched with a dog. They are trained to sidle up next to their person to provide a little bit of support and leaning ability as well, to kind of really support. They are also, many of them, trained to pull the wheelchairs and assist in that kind of mobility if necessary as well.
You use the terms matched with the dog, so I think the question then would be, so an animal, when he or she is chosen to go to a specific person, they are specifically trained for the needs of that person?
Yes, so all of the dogs have a standard set of training commands placed into their brains and into their drive. But when it comes to the matching process, actually it's kind of a date. So the individual is called up and told that Canine Companions has 1, maybe 2, or 3 dogs that they think might work. And during the first couple of days of team training, that's what we call it, we match them and have them, so to speak, date for little rendezvous, little tasks, and see what kind of pairing presents. And the trainers and the professionals who know these dogs very well can watch that dog behavior, see the bonding begin, and choose the right dog for the life of that partnership.
So very specific thought process and training processes going into that for the benefit of the dog and the eventual owner.
Absolutely.
You might share with our audience who might be listening anywhere across the country, if I were in some other part of the country and I thought I had a need for a companion animal, and I'm just hearing this today, where would I go to start that process? Is there a website? Is there a phone number? Share with us how we might do that.
It's as easy as canine.org, and we should spell that out. It's canine.org. And on that website, you can look up the different types of dogs that the organization has in their training process. You can even learn about all of our research that we are doing to make sure that we're training the right healthy dogs for the life of their work and for the benefit of canines. But you can also see all of our regional sites and which site your city and your town would be filtered to. Because in the state of Florida, in the 8 most southern states, we go to the southeast region. But out in Texas, you go to the southwest region. There's also the northwest, northeast. We have them 7 different locations across the country.
So once an animal becomes a partner with a person, how long does that dog function as a service animal? Is there a certain length of time?
There is no specific certain time that a dog will work for someone. It is a conversation between the dog handler, the person that was matched with the dog and canine companions, and the veterinarian, of course, because dogs will age and dogs will tire depending on how hard they're working in their daily life, and perhaps whatever illnesses they may inadvertently or unfortunately come down with. We do a great job of genetic testing our dogs to match and breed the right healthy matches, so that we have the healthiest of dogs that are out there. But it's between 8 and 10 years of age, maybe longer. But once you're with canine companions, you've got us for the rest of your life. So there's a conversation that starts up when we're talking about retirement for your dog. Are you ready for us to start talking about the next dog? And should we send you our application for a successor dog, is what we call it.
So kind of an emotional process for that owner and for the dog as well. They've been together for a number of years, taking care of each other, kind of fallen in love as owner and pet, but a great service for the person who needs that help. We've had a number of our members, I know 2 of our board members who have had service animals, and we as a board, we fell in love with those animals as well because we appreciate the personality of the animal. As you watch that animal take care of the person they are trained to take care of, it's so impressive to watch, and you know the life of that person is enhanced greatly because of what that animal can do and will do for them.
Yes.
Leah, is there anything else that I should have asked you about this process that we've left out that you think we should communicate?
Well, I think it's kind of important, or just a little side note. I'm coming to you from Canine Companions, but I think I should share that I too have HSP. I have SPG3A, and while I come as a representative of Canine Companions who has a facility dog, and I work in a children's hospital with one of their wonderful dogs serving multitude of children, my goal when Brea and I retire from our work life is for me to get a service dog from Canine Companions myself, because I know what she has done for me inadvertently, even though she's not my service dog. I know that I need one for the rest of my life.
Well, we thank you for being here with us today in Canine Companions, and you just hit on another storyline here that I want to make sure our listeners and the folks who watch this in the future don't miss, and that is that as we work to put this together here in this area, we had no idea when we started, when we reached out to the Tampa community and asked for help with our children's program and help with what we're doing here. We had no idea we were going to meet a fellow HSPer who deals with HSP. And so we appreciate meeting you in that regard. We want to recruit you in your involvement and support with our foundation as well. But thank you for being with us.
Absolutely.
And thank you for sharing with us. And if you're listening and you think you might have a need or want to consider one of these beautiful animals to assist you, Miss Leah has told you how to do that, so get online and start that process. And if you hit a snag, we'll find someone to help you. So Leah, again, thank you so much.
My
we appreciate
pleasure.
your time.
Thank you.
Well, guys, we've had a great day. We're about to have another session in here in just a moment that you'll be tuned into as we talk about the issue with child onset issues with Dr. Fakari and some of the parents in our group who are dealing with that. So take a break for just a moment or two, and we'll be back with that group to close out the day. When we finish that session, we'll be finished for the day, getting ready for dinner here, so we'll let you go. After that, we'll see you in the morning about, I think it's 8:25. You got the program, so we'll be with you about 5 minutes before that program begins, and we look forward to this last session and to tomorrow. So thank you for spending the day with us, and we'll see you in the morning.